Welcome back to our class. Don't forget to subscribe if it is your first time to visit our channel. Another disturbing question to students in paper 1 of life science. The development of the zygote until implantation. Students really struggle with this question. But before this video ends, you'll be able to write what is needed in the paper, and then you score all the marks. So development of the zygote until implantation. Let's get started. Let's first describe all the stages which are involved in this process. So you start with the sperm and the oocyte. Oocyte is the ovum. So you start with the sperm or spermatozoa, fuses the nucleus of the sperm fuses the ovum, and then when it fuses, it forms what's called a zygote. So it means that here you have the nucleus of the ovum and then you have the nucleus of the sperm. It's not the whole sperm entering, no. It's just the nucleus of the sperm. That's why we are saying that haploid nucleus of the sperm, not the sperm entering, no. Haploid nucleus of the sperm fuses with the haploid nucleus of the ovum to form a diploid zygote. So a zygote is one cell but is diploid. It has two sets of chromosomes from the father and from the mother. While the sperm is haploid, has one set of chromosome, and then the ovum has one set of chromosomes. So this is haploid, this is haploid, half plus half, you form what's called a diploid. So the zygote must undergo a process which we call mitosis to form different cells. It undergoes mitosis, like one cell divides to form 2, 2 divides from 4, 4 divides from 8, like that. So it undergoes mitosis repeatedly. So it forms what we call a ball of cells. So when it undergoes mitosis repeatedly, it forms what we call a ball of cells. We call it morula. This is how a morula looks like. It's like a ball. That's why you say that it forms a ball of cells called morula. So we are saying that this ball of cells undergoes mitosis continuously, forms a space inside. This space, so it becomes hollow. It means that it has a space inside. So it forms a hollow ball of cell called blastocyst or blastula. Yes. So it was a ball, but now the ball forms a hollow, forms a space inside. You can see it here. See, the space inside is big. So this space, it makes it to be a hollow. So it forms a hollow ball of cells called blastocyte. So we are saying the blastocyte now, it forms the outer layer. This outer layer, yes, we call it now the chorion. This outer layer starts to form the finger-like projections. These are the finger-like projection, which we call the chorionic villi. These chorionic villi are the ones which are very important for implantation. So basically, that's how you can explain it. So we are saying that how do you write this in an exam? The sperm paralyzes the ovum to form a zygote. So we can write that after fertilization, the zygote undergoes mitosis. So this zygote, it undergoes mitosis. When it undergoes mitosis, it has to undergo mitosis. One cell divide, form 2, 2 forms 4, 4 form 8. So it forms a ball of cells. So we are saying that it forms a ball of cell. This is a ball of cell called morula. So the zygote, yes, undergoes, we give you a tick, undergoes mitosis, we give you a tick, to form a ball of cell. This ball, you have to write it that a ball of cells. To form a ball of cell called morula. So we give you a tick, a ball of cells called morula, we also give you a tick. So that's how you try to, each statement which is in different color, it shows that that is a tick in the exam. So it depends on how the question is set. But in most cases, if it is an open question, then it means that each color is a tick. The morula still undergoes mitosis. When it undergoes mitosis, then we are saying that it divides by mitosis repeatedly to form a hollow ball of cell called blastula. So if you look at it here, yes, it undergoes mitosis repeatedly. So to form a hollow ball, it has a space, as we explained it before, hollow ball of cell called blastula or blastocyte. So we are saying that it forms the outer layer called chorion. So if you look at it, yes, this ball of cells, yes, the outer layer, this outer layer, yes, this outer layer, it forms this outer layer which you call the chorion. So this chorion, it modifies, it forms a finger-like projection finger-like projection, we call it chorionic villi. Yes, it is a chorion, but now the finger-like projections, which we call chorionic villi, are used for implantation in the uterus or the endometrium. So basically, this is how it looks like. This finger-like projection forms what we call the finger-like projection, which we call the chorionic villi. So this is the outer layer is the chorion. So this outer layer starts to protrude out. These protrusions or the finger-like projections, call them chorionic villi. These chorionic villi are very important for implantation. So basically, that's how you need to explain it. Let me just repeat it once and for all. After fertilization, yes, when they ask you to explain this, don't explain the process of fertilization. We don't need to know the process of fertilization. They want you to explain what happens to the zygote immediately, or what happens to the zygote until implantation. So you say that after fertilization, a zygote is formed, which undergoes mitosis. It undergoes mitosis to form a ball of cells, called morula. Morula undergoes mitosis, or it undergoes mitosis repeatedly to form a hollow ball of cells called blastula. So it forms a hollow ball of cells called blastula. It forms the outer layer called, this outer layer forms the outer layer, which we call the chorion, which forms the finger-like projection. So it develops the finger-like projection, which we call the chorionic villi. What's the function of the chorionic villi? It's very important for implantation. So basically, that's what you need to know, nothing else. So just have to know that this is a zygote. This is an ovum. It is haploid. This is the sperm is haploid. The first cell is a diploid, because haploid from diploid. This is also a diploid. Yes, this is also a diploid. Also, this is a diploid. So basically, these are undifferentiated cells. They can develop into other structures. If you look at it here, it's just trying to show you the 3D presentation. That this is the egg. Yes, the egg after being fertilized. What happened? The egg immediately after fertilization. So yes, this is the egg. It undergoes mitosis repeatedly to form a ball. Yes. From here to here, it is all that is mitosis happening, which you call morula. Morula undergoes mitosis to form a hollow ball. Inside there is a space, hollow ball of cells called blastula. Then blastula, yes, forms the outer layer, which you call the chorion, which forms the finger-like projection. Yes, finger-like projection for implantation. So basically, that's it. And then here are some of the questions. Identify the process taking place at 1 in the diagram above. So if you look at it, sperm fuses the ovum, so it is fertilization. State the type of cell division that takes place in 2. You see, we talked about it. So the type of cell division is mitosis. Then there's a give 2 functional of extraembryonic membrane produce functional extraembryonic membrane. So we say that it forms a chorion. Chorion, yes. Then also we have what we call the amnion. So we have not talked about amnion because we have not yet reached there. So it forms the chorion and then the amnion. Identify the stage that of development indicated by A. So A, stage of development, this is just a zygote, and then B is a morula, and then C is a blastula. Name the part of the female reproductive system where events in the diagram above usually takes place. Fallopian tube. Look at it here. It occurs in the fallopian tube. So this is how the sperm fertilization takes place here. So this is the development. So implantation occurs here, exactly here. So this is the ball of cells, and this is the hollow ball of cells. Within a maximum of 1 week, this process has taken place. This is the time when a person has started to show the signs of pregnancy. Someone is weak, someone is vomiting. This is the period. Why? Because the body is experiencing new things. So we can also set it in this format, and then we ask you what is really happening here. Describe the process, you see. So until implantation. From this point after fertilization, then it goes until it reaches here. So next time we will look at fertilization in detail, and then I will show you how we can also write it without having any problem. See you next time. Subscribe if you have not yet subscribed for the new videos.
On what we call degrees. Why? Because the circle is made up of 360 degrees. So you cannot plot it in you cannot plot it in percentages, mass, what must be in degree. The first thing which you need to have is a protractor, because there is no way how you can draw a pie chart correctly and then when you don't have a protractor. The protractor has two scales. The first scale is the clockwise scale. The second scale is the anti-clockwise scale. There is this scale which starts from 0 here, and then it moves like this, comes here to 180. And then there is another scale, outside scale, comes from here, it moves, then it comes, it's 180 is here. So it depends on whether you are a left-handed person or right-handed person. The two options are there. Oh, you can be even left-handed and then use the other scale. So it depends on what you really need. So once you know how to use, so I usually call this one, because it's moving anti-clockwise, the anti-clockwise scale. Outside one, usually call it the clockwise scale. So any scale you use, still you get it correct. The second thing you need to have is you need to have a compass. This compass, a pencil must be well fit. You must fit it properly into the compass. Then it has you must have what called a ruler. So the function of the ruler, the first function of the ruler is to measure at least 5 centimeters. The distance between the first tip, the tip of the compass, and the tip of the pencil must be 5 centimeters at least. Why? So that you can draw a bigger circle. Eh? Yeah, we shall see how we draw it. If this space between the tip of the compass and tip of the pencil is too small, for example, is here, the circle is going to be very small. Yeah, it's going to be very small. So this makes the work to be difficult to be marked. So the more you draw it bigger, the more clearer it becomes. So it also makes the marker happy when it's marking, because it's not going to strain the eyes. And so these are the steps we're going to be doing until we completely draw the circle. We're going to see that practically, or what you call the hands-on. Then you are given a table which has all the information which you need to use for plotting. So sometimes this is not added, sometimes it's added. If it's not added, so you have to add them. For example, the total gonna be equal to 18 plus 9 plus 6 plus 3. So if it gives you 36, so these are not degrees. It depends on what they have given you. Yes, if it is in degrees, then you just plot, just do the pie chart without converting. But if it is in a mass, number of people, what, percentages, you have to convert them. So to calculate for the 18, I'm going to say 18 is blue. Blue is going to be equal to 18 out of the total, which is 36 times 360, so that I convert it into degrees. So I bring my calculator. Then once I bring my calculator, it's going to give me 18 divided by 36, answer times 360, I think it's 180. So this is going to be 180 degrees. And then I go to the next one, is green. Green is equal to, what is it? 9, eh? 9 over 36 times 360. You bring your calculator again, and then you say 9 divided by 36, answer times 360, it's 90. These are down, these are the degrees you're supposed to plot. The degrees. And then you also do for red and then yellow the same thing. And then end up having this. You also end up having that. Then once you have got these degrees, then automatically it means that you are ready to go for drawing a pie chart. So let's go for drawing the pie chart. You need to have a compass, a ruler. This is called a protractor. Yes, you need to have a pencil, and then you need to have a pen. So these are the things which we are going to use because we have already done our calculation. So these are the things we are going to use so that we can start drawing. So the first step is to put the this. If it doesn't go through, you open. Please don't go to the paper without having this. So you make sure that they're in on the same what? Same level. Yeah. You see that they're on the same level. Yes. Once you see that, yes, they're on the same level, then you make it tight so that it doesn't move and doesn't shake. All right. So that's number one. Then step two is you're supposed to measure. Remember, I told you that try to measure this, measure 5. So 0 to 5. The reason why I'm saying it is good to measure 5 centimeters so that when you draw, for example, you want to draw it here, you make this one tight, and then now you turn this like this. Make sure that the center doesn't change, because if it changes, yeah. So you see how big it is. It's big enough. If this is small, is smaller than 5, for example, is 2 centimeters, you will end up drawing something very small. See? Something very small. And then this is going to give you a problem when it comes to measuring the degrees. So we prefer you to use 5 centimeters so that at least the circle is big enough. And then it also makes the examiner happy. It makes the examiner happy when you are at least. So draw it, put it there where you want it to be, and then now start to draw. You can even just do like this, just change the paper like this. You change it correctly, yes, it will give you the correct circle. Or if you don't want to change the paper like that, you just draw, keep it, and then start. Make sure you use a sharp pencil, yeah, because we want some things which are sharp. Yes. So we want things which are sharp. So I'm done. That is the first step which you needed to know. That is the first step. So now the function of the compass is done. I can remove my pencil. Yes. If you see that this is not sharp enough, try to be with your sharpener just next to you. Try to make it sharp. Yes. And then now draw. You have to be neat. You have to be neat. So you draw a line going to the circumference. So it means that I've drawn what you call the radius. Now I'm going to bring my protractor. This is a protractor. I said that this protractor has 2 scales, has a scale from 0 to 180. So I will say that because it is moving anti-clockwise, Therefore, I'll call it the anti-clockwise scale. And then this one from 0 to 180, the outer one. So I'll call it the clockwise scale. Yes. So there is this part. Yes. This part is where it's supposed to be placed onto the, you're supposed to place it where the compass was. And then this line is supposed going through this line you have drawn. So I put it there. I put it there. Yes. So I see that now it is in line. So now I come back here and then I check. The first degree is 180. So I come here and then I measure. The first degree is 180. So I come here, it must be straight. So now if I use, because now I've started from this side, I'm going to use the inner scale because now I can't measure from that side. So because this is my first point, yes. So I measure 180 is gonna be there. So I put a mark there. Yes. So now after that, I bring this ruler and then I join it. I join it to the mark. So I join it to the mark. So basically that is 180. But because now I stopped here, so now I have to turn this paper. And then I look for the second one. The second one is 90 degrees. So now I do the same. Remember, this is our center. Yes. Now I put it here. Yes, I put there. So because I'm using anti-clockwise, you can use even clockwise. You can measure from that side, depending on your choice. So I come here, say 90 is here. Put it there. Then now I come and then I draw 90 is there. So it means that this is 90. Then I come to 60. Then I turn it. 60. Measure 60. Yes. 60 degrees. 60 degrees is here. Degrees is there. And then now I come here. I use my ruler. And then I draw 60 degrees. So, yep. Now, the remaining one must be 30 degrees. If it is not 30 degrees, it means that you did something wrong somewhere. So if I check here, you see that 30 degrees, it clicks in 30. You see, it clicks in 30. So basically, that's how you supposed to draw. Now, you can use a compass or you can just use hands, and then you tell us that this are those, how many degrees are those, then how many degrees. You can use a compass so that it looks, yeah, it looks nice. And then say that I need to know what is a key. Then I say, now I start to do whatever I'm doing. Maybe I call this one. Let me just shade it like this. Yes. Let me shade it like this. I come here and then I say what I've shaded like that. Now I've shaded it like this. And then now I come here and say that this is the blue. So this is the blue. And then I come here, maybe I call this one S, 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 S. Come here, the box and say S, S, S means what? S, 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 it is S, 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 it is the smallest. So this is 180. Yeah, 80 degrees. This one is 30 degrees. This one is was 90 degrees, and then this is 60 degrees. So basically, that is it. This is it. S is yellow, and then I can call it P, whatever you use, or whatever color you use. And then this one, PPP, is the 60. We said is red. Then say this is red. Then say PPP, PPP is red. And then lastly, maybe I can call it XX, whatever you sign use. And then I come here and then I say XXXX, and XXX is what? XXX, XXXXX is going to be is green. XX is going to be green. So basically, that is it. Now, the next thing is you're supposed to write the caption or the title. Don't forget to write the title. The title is supposed to have the two variables. So now, how do we mark? Basically, yes, we will mark the type of the graph. It's a pie chart. Maybe give you a mark, and then we'll mark the caption. We'll give you a mark. We'll mark the plotting. We'll give you maybe 2 marks. Yes, we'll mark the key. We'll give you a mark there. We don't stop there. Then we come back here and we see the calculations you have done. Also come here on the calculations. Maybe we shall say that calculations also, maybe we give you 2 marks for each. And then now maybe they say calculations, 2 marks. Then depending on the memorandum, how is the memorandum, what is the memorandum saying? So basically, that's how you're supposed to draw a pie chart. Let me know your problems so that we can solve those problems, because this was one of the questions for one of the students, say that wanted to know basically how do we draw a pie chart. Thank you very much.
Feedback mechanism, but we don't talk about it here. When you go to the universities, you'll talk about it. But now here, when you talk about this negative feedback mechanism, it means that something which went wrong must be brought back to by subtracting what that made it wrong, then you bring it back to normal. That's why we call it negative. But if something get wrong and then it becomes more wrong, then it becomes positive feedback, but we don't talk about it. Yes. So here, this is just to bring about the normal functioning of the body so that everything works to the way it's supposed to work. So this enables the body to function efficiently. So when it works efficiently, there is a difference between efficiency and effective. So don't use effectively, use the word efficiently, because when you talk about efficiently, yes, the body is supposed to work. When you say efficiently, sometimes the efficiency might not be about 100%. When you go to physical science, you know that better. But despite the changes which might happen in internal and external environment. When you talk about external environment, we're talking about the surrounding areas. What you have, maybe the temperatures of the surrounding, maybe you talk about the humidity, that's the surrounding. When you talk about internal environment, we are talking about maybe the blood glucose, the salts, the water. Did you know that a human being has the same temperature? However much this person goes to water or to the ice, the temperature remains the same. So that's what you're saying. Despite all the changes which might happen, you understand? Yes, the internal environment must be kept constant. So now, if it fails to be kept constant, then now the problem will come in. How does the problem come in? Maybe the system will fail to work properly. I think I'm clear there. Good day, sir. Hope you are well. Thank you very much for simplifying lessons, making it easier for us. Okay, thank you very much, guys. Yes. Make sure that you do what? You share to others also so that they can benefit. Guys, if you have a problem, this lesson is live. Ne? This lesson is what? Is live. So if you have any problem, let me know. I can try to answer those questions. I'll be giving you some questions. This lesson might go for 1.5 hours, if not 2 hours, so that we maximize all the things which you might make this situation to be difficult for you, so that you guys will get distinctions, because that's what we are looking forward for. All right, we are saying that changes in temperatures, glucose levels, carbon dioxide, and water levels, plus salts, these are the internal environments we are talking about, which are supposed to be kept constant. Yes, so those are the internal environment we are talking about. So this internal environment must be kept constant. Yes, so we are going to look at them in detail. So they are saying that regulation of glucose levels. Now, here you need to understand some few things before you go to the regulation. Let me give you a trick so that you can understand this better if they bring it in terms of this diagram you see. Let's start with the glucose. Number one, glucose. Guys, you need to know two things from two things. The first thing you need to know is always in the middle here, you have what called the normal. Guys, all of the mechanisms, they look like this. You don't need to stress. So always it is like this, normal. You understand? When you say normal, okay, what is going to happen here? The last statement and the first statement must be in opposite way. If you see here there is increase, yes, the last statement must be decrease. If you see here there is decrease, yes, the last statement here must be increase. I think I'm clear there. Whatever negative feedback mechanism you're gonna talk about, except one, that is carbon dioxide. I'll explain that better regarding to why carbon dioxide is not made to be like that. It's because its mechanism is one way. It's not two ways like these ones. So now we are saying that the first one must be, if it is increase, the last one must be decrease. If the first one is decrease, the last one must be increase. So now you only have to fill in 2 points here. So make sure that you separate these two from these two. So how do I know these 2 points? You need to know what are they talking about. This is also being spoken about here. The normal level of glucose in the blood. So if glucose level increase, the last statement must be glucose levels will decrease. If the glucose level decrease here, the last statement here must be the glucose levels increase. I think I'm clear there. Now, when it comes to glucose, then you have to ask yourself, when it comes to glucose, which gland is responsible for glucose? And what is the function of that gland? So you mean that if you're talking about glucose, glucose goes with glycogen. So glucose, yes, is being acted upon by which hormone? And which gland does it produce that hormone? So it means that the hormone and its function will be here, and the gland and its function will be here. Meaning that the function of the gland is the hormone it produces, and then the function of that hormone will bring about the decrease in that level of whatever you are talking about here. For example, say that glucose level will increase, or definitely the last statement will say that the glucose levels will decrease. So that one is sorted. 2 marks gone. Oh, you have got them before even you go to the paper. Then you have here pancreas, because the glucose, what works on glucose and glycogen is pancreas. So the pancreas, that's the gland, will produce or will secrete insulin, meaning that you have talked about the gland and its function, meaning that the gland, which is pancreas, what is its function? It secretes insulin. Done. Then you come here and you tell us the function of insulin. This insulin, what does it do? You tell us that now insulin will stimulate the conversion of glucose to glycogen, because glucose is too much. So insulin will stimulate the conversion of glucose to glycogen. My students, please don't say that insulin will convert glucose to glycogen. We'll get it wrong, because insulin does not convert glucose to glycogen, but insulin stimulates the conversion of glucose to glycogen. What converts this is the liver, because when you go to the university or other areas, you'll find out that we'll include here a liver. Yes, we'll include isolate of Langerhans. When you are specific to the, then you talk about the beta cells, the alpha cells. We will talk about those things when you go there. But for now, what is most important is, you know that insulin produced from the pancreas, then that insulin is going to stimulate the conversion of glucose into glycogen. Here, that's why here I wrote that glucose is converted to glycogen. What converts it is the liver. What stimulates the liver to convert glucose to glycogen is insulin. So basically, you just know the gland, and here which hormone does it produce, and then here you come here, you talk about the function of that hormone. That is the stimulation. So basically, that's what you need to know about it. So when you go here, it's the reverse. It's the reverse. So you don't need to get stuck when you reach there. You understand? Does Sir offer lessons or videos? Yes, the lessons are there. The lessons are there, but unfortunately, I'm not a math teacher. I'm a life science and physical science teacher. So it means that I only offer here life science and physical science lessons, but maths, other subjects, all other subjects are here, but by different teachers. Yes. So if you have questions, still you can drop them in the comments. Any video you comment on, still we will bring your response, or we will try to act on your question you have answered, you have asked. Anyway, let's continue. Let's go to what about when there is a decrease. If there is a decrease, yes, it means that glucose level has decreased. The last statement must be glucose level will increase. When glucose level decreases, talk about the gland. So which gland is the same? Pancreas. Pancreas is going to secrete glucagon. Don't forget that you have glucose here, glucose, and then you have glycogen. So now here you have, if you want to convert glucose to glycogen, use insulin. And then if you want to convert glycogen back to glucose, you use glucagon. Yes. So now it means that now the pancreas will secrete, because now we want more glucose, because the glucose levels are low. So the pancreas is going to secrete, yes, glucagon, and then the glucagon will stimulate the liver to convert glycogen back to glucose, and then you go back to normal. Guys, this is the same situation when you have, for example, you are hungry. It's break time, you are hungry, and then after that, They don't tell you to go to maybe the teacher say that you guys, you have to complete this. Then after a certain time, the hunger will go. But how did that hunger goes? It goes because now the glycogen has been converted back to glucose, and then now the body will receive this glucose and it will become normal, or it will become okay again. And then life continues. Yes. So basically, the storage form of glucose is glycogen. The storage form. In animals, the storage form of glucose, if they ask you that question, what is the storage form of glucose, is glycogen. The usable form of glycogen is glucose. The usable form of starch that is in plants is glucose still. So you have to know that, just so that if they bring the question, you are able to answer such questions. Now, this is the same thing I've explained. You don't need to memorize, like, no, cram. You just need to understand. First step increase, last step decrease. Which gland? Okay, you know the gland. Which hormone produces? You talked about the hormone. So what is the function of that hormone? So therefore, you have got to know the function of that hormone, and then after that, that hormone is going to bring about a decrease in the level of glucose, and then the situation goes back to normal. Glucose level in the blood increase, yes, above the normal level. The pancreas is situated in the is stimulated to secrete insulin into the blood. So what is the function of the? You see, it goes above normal. You talk about the gland. What does the function of, what is that gland do? It produces insulin. What is the function of the insulin? This one stimulates, you see, stimulates the liver to convert excess glucose to glycogen. You see, once excess glucose has been converted into glycogen, definitely the level of glucose will decrease and goes back to normal. That's what I was trying to explain here, but here is just in words. And then here, glucose level, decrease below the normal. So what happens? The pancreas, you talk about the gland. What's the function of the gland? Produces glucagon. What does glucagon do? The glucagon will stimulate the conversion of the stored glycogen back to glucose. The moment glucose is available in the blood, definitely glucose levels in the blood will increase, and then it goes back to normal. Simple. Simple. Guys, let me know. Is this thing simple or is that difficult? Ne? I don't think these things are difficult. You just need to put love. Some people, they call it love. Yes, put love there. Yes, the way you want it to call it. And then after that, you will see that these topics are very simple. Yes. Let's look at some of the abnormalities or the deficiencies here. So I've explained this. I've explained that, and then yes, Young Seri is saying that you make these topics easier for me. Thank you very much, Young Seri. And I always see you whenever I do my lives. You are always there. So I credit you for that. Ne? Thank you very much. Yes. So let's continue, guys, to the next question. Yes. All right. Let's go to the next sub section. So here you have glucose. You have glucose. Here you have glycogen. From glucose to glycogen, you use insulin. And then from glycogen to glucose, you use glucagon. Simple. So what produces this is pancreas. And then once you know, guys, go to the paper when you know this. Ne? And then number 2, you know how to manipulate this. Simple. Once the moment you know how to manipulate this, then you will be able to know all this. So we're going to see some of the questions which are concerning about this. You'll see that the questions are simple to answer. Diabetes. Some of the conditions which you find in this glucose situations is diabetes. If the body cannot produce sufficient insulin, if the body, the pancreas, cannot produce sufficient insulin, you're going to suffer from what called diabetes. My students, please don't say diabetes. Because when you say diabetes, we will cross it. If the pancreas fails to produce sufficient insulin, insulin, then you're going to suffer from diabetes mellitus, not diabetes. Because when you say diabetes, we want to know which type of diabetes are you trying to mean? Because we have two types of diabetes. We have diabetes mellitus and diabetes insipidus. The diabetes of sugar and diabetes of water. So now, here, diabetes of sugar, diabetes mellitus, is divided into diabetes type 1 and diabetes type 2. Please don't say sugar diabetes. No, say diabetes mellitus. Simple. Don't complex your life. You bring in, what if I say this? What is the problem for you to memorize diabetes mellitus? Why do you want it that way? Yeah? Why do you want that way? This is what we want. Don't complex your life by losing a mark. Just say simple diabetes mellitus. Simple. You understand? Yes, you go to the paper, come out when you're happy. Don't come out of the paper when you're happy, when you're writing diabetes instead of diabetes mellitus. Let's continue. They're saying that the pancreas is not producing hormone or hormone that is insulin, meaning that insulin, you know that insulin is very important for stimulation of the conversion of glucose, the excess glucose to glycogen. So automatically, if there is insufficient insulin, it means that excess glucose will not be converted to glycogen, and then now the body will have a lot of glucose, and then this glucose, the person will start to suffer from diabetes mellitus. How do you test for this diabetes in a simple way? Yeah, if the amount of if you go to you urinate, and urinate, you find glucose in your urine, that could be some of the few signs that maybe diabetes is coming. I'm not saying that if you find it, you are having it, but it could be the sign. Yes. So we are saying that diabetes type 1, what happens? The pancreas is not producing hormone, that is insulin, necessary for controlling the glucose level in the human. Diabetes type 2, that is diabetes mellitus type 2, in other words. Yes, insulin is available, but the body cell is unable to control the what? The glucose. So you find, we can bring diagrams like this, and then we ask you what is being illustrated in the above diagram. Yes, so that condition is diabetes. Here is a question. Here is a question, and the question is asking you to give us some of the answers which are here. Can I do bingo for you? Can I do bingo for you? Meaning that, can I give you a few seconds to do them, to do the questions, and then, okay. If you are on live, I'm giving you 1 minute to answer. 1 minute. 15 seconds. Yeah, 15 seconds. So that we go through and we see that what we went through or what we have gone through is correct. What you have done is correct based on what we're gonna give as the answer. Okay. 10, 8, 6, 4, 2, 0. Okay, let's go through it. Let's go through it and then we see. Okay, you're saying that you have high level of glucose in the blood. This level, yes, there is a pancreas here. Why is this organ called pancreas? Yeah, don't forget that a pancreas looks like a banana. I mean a real banana, eh? The banana for gorillas. You know, gorillas and chimpanzees like bananas. Anyway, then low levels of blood glucose, yes, but here it's still something is going to the pancreas, and then now this release of hormone 3, release of hormone 2. See, and then now you have increase blood glucose, and then here decrease blood glucose. Here comes a question, and we say organ 1. What is organ 1? We have seen it that organ 1 is pancreas. Hormone 2, where is hormone 2? You have high level of glucose. What do we need? We need, you see, the last high. If there is an increase in the level of glucose, the last statement must be decrease. You see what I say? Yes. Increase, it means that there is more, because here they say the high level of glucose. The last statement here is low level. Why am I comparing this? Because this is dotted, it comes like that. Yes. Meaning that now the blood glucose go back to normal. So if there is high level of glucose, we need a hormone which is going to make it decrease. So which hormone is that? Is insulin. Insulin. Meaning that insulin, hormone 2 is insulin. Definitely. Hormone 3 is what? Glucagon. Because if it is not insulin, it's glucagon. There is no any other hormone which is going to be talked about in regulation of sugars. Yes. So, meaning that hormone 3 is, okay, is glucagon. They're saying that. Guys, there is a confusion between glycogen and glucagon. Glucagon. Glucagon is a hormone. Glucagon is a hormone. Ne? And glycogen is a substrate, is a food. Anything which has this O becomes a food, like sucrose. Sucrose. When you say sucrase, it becomes an enzyme. So it's like a hormone. It's the same thing. You talk about, maybe you talk about lactose or lactase. So when you have A, is a hormone or enzyme. Ne? A. So glucagon. I don't know whether you read it as glucagon. I don't know. Okay, doesn't matter. Glucagon, if there is A, that's a hormone. Where is O? O means food. Don't forget that, guys. Food. O, food. Food. So you just know that this is not a hormone, this is a substrate. This is a food. Or so guys, I think you no longer now messing it up. You're no longer messing it up. When are you going live for grade 11? Soon. I think on Friday and Saturday. I'll be coming for excretion. Yes. So we are saying that they are saying that disorder caused when gland 1 fails to release sufficient amount of hormone 2. So we don't have any disorder here. The only disorder we have is diabetes mellitus. Simple. The mechanism, when they talk about the mechanism Then you talk about negative feedback mechanism. When they talk about the condition, then you talk about homeostasis. So basically, homeostasis is a condition of the internal environment being kept constant. But what makes it to be constant is the negative feedback mechanism. I think I'm clear there. Is that difficult? You must remember. Ne? Yes. We will do more questions towards the exams. Ne? Towards the exams, as we are planning to be live for 24/7 from the beginning of prelim for all subjects until you write your end of the last question, the last paper in your final exam, meaning that you will be live for almost 2 or 3 months. Yes. So keep yourself on Thunder Educ so that you can obtain a distinction. All the help will be here. We are saying that the mechanism that controls the level of glucose in the blood, we say that that mechanism is called negative feedback mechanism. Negative feedback mechanism. Think I'm clear there. Is there any question before I go to regulation of carbon dioxide in the body? Regulation, guys, if you have any question, let me know in the comment section. Ne? Yes, if you are looking for the distinction material book, yes, I've sent the link there. Yes, you'll find for grade 10, grade 11, and grade 12. And many books are now, we are trying to write them so that you can also benefit. Unfortunately, most of them might be out next year. But for grade 10, 11, and 12, life science, they're already there. They're uploaded. You can find them by following that link in the description below. All right, let's go to regulation of carbon dioxide in the body. Regulation of carbon dioxide in the body. How is carbon dioxide regulated? Guys, I told you that for the case of carbon dioxide, it only has one way. It doesn't have two ways. It doesn't have two ways. Let me check here. Let me check here before I go to. Okay, in this one they brought a thermal regulation. Did they? Okay, this one I'll talk about it. They bring about no. Okay, I'll talk about. Let me first describe everything. Then I'll go to the questions separately. So here they're saying regulation of carbon dioxide in the body. What are you talking about here? Here, you need to know. Is carbon dioxide important in our body? No, we don't need carbon dioxide. So, any level of carbon dioxide is not needed. Therefore, we have to remove out of our body. That's why carbon dioxide level in the body only has one way. It doesn't have two ways. No, it only has, if you check this one, yes, you'll find out that it has the upper one and the lower one, but carbon dioxide only has one way. Decrease in the level of carbon dioxide, that's better. If it's not there, that's good. Increase in the carbon dioxide level, we don't want it. That's why its regulation is related to decrease in the amount of carbon dioxide in the blood. Okay. First of all, carbon dioxide is supposed to be detected. When you say that, number one, someone asking me, where do I start? You start from here. I put it number one. Stimulus. Stimulus. What's the stimulus? Increase in the carbon dioxide level in the blood. And this leads to decrease in pH and decrease in oxygen. Why is oxygen decreased? Because we use oxygen in a respiration and we produce carbon dioxide. That's why it means that if I have low level of oxygen, it means that I have high level of carbon dioxide, because oxygen has been used to produce energy, and the carbon dioxide is a byproduct of respiration. Now, how is pH decreased in the blood? How is that linked? When we look at it scientifically, sorry guys, I'm just going to use just a small bit of chemistry. Yes. We know that. No one is going to ask you this, but I'm explaining to you how did this low pH come about? And how is it linked to carbon dioxide? Why do we need to talk about low pH when you're talking about carbon dioxide? Yes, when carbon dioxide reacts with water, don't forget that our body is above 75% water. So we have water. Yes. So when carbon dioxide reacts with water, you form, this is a weak acid. It's a reversible equation. Okay, you form what you call carbonic acid. We call this one carbonic acid. The best example of carbonic acid is acidic rain. Rain which is acidic. Yeah, when now the first rain you're gonna receive after winter, because it has stayed longer without raining, so carbon dioxide has accumulated in the atmosphere. When that water comes and reacts with the carbon dioxide, however much is not a good, it does not dissolve in water easily, but there is some bit of dissolving, so it forms that acidic water, so acidic rain. So in this case it forms carbonic acid. So it means that now the blood, because this is water, the blood becomes a little bit of acidic. And if you have acidic acid, it means that the pH decreases, because from 1 to 6, it is acidic. So it means that the more it goes down, the more it becomes acidic, the pH becomes lower. That's why you say that increase in carbon dioxide level decreases the pH. You understand? So meaning that whenever I have too much carbon dioxide, the pH is going to be decreased. Because our receptors in the body will not detect carbon dioxide, but will detect the pH. That's why, number 2, receptors, low pH detected. You see, how does it detect? Being detected is being detected by this carbon dioxide reacting with water to form carbonic acid. Then this carbonic acid is being detected, because carbonic acid is a chemical, is being detected by the chemoreceptors. The receptors in the body which detect these chemicals. So these chemicals are being detected by chemoreceptors. Am I clear? Am I making you lost? When is prelim section live starting? We are preparing the gadgets, yes, for helping us so that different teachers can streamline at the same time. Yes. So maybe by the end of this month, maybe around there 2020 something. Yes. The lives will start. Will start, so that now it is 24/7. Anytime you want to check, or if you ask a question, maybe from WhatsApp or what, we switch to it, and then we prepare for you, and then we answer those questions immediately. All right, let's continue to, I think I'm clear there. So number 1, when you go back to what is needed in life science, increase in blood carbon dioxide levels decreases the pH, or lead to the decreased pH, and then the decrease in oxygen. I think I've explained that. And then now, number 2, the receptor will detect the low pH using, what are they going to use? The chemoreceptors. We call them chemoreceptors because they detect the chemicals. Yes. Number 3, now the moment receptors detect, what is the next step? The receptors will detect, and then they will have to send an impulse. So these receptors, remember that receptors always the sensory cells, basically. Yes, when you go to the nervous system, the sensory cells will change this message of chemical into a nerve impulse. So impulse is sent to medulla oblongata. Why not cerebrum? Why does it go to medulla oblongata? Question. Why? Okay, it goes to medulla oblongata because medulla oblongata controls all involuntary actions. Not reflex actions. There is a difference between involuntary actions and reflex actions. Actions which are done to protect the body from danger, we call them what? We call them reflex actions. But actions which work without control, but they are not linked to protection of the body, then we call them involuntary actions. For example, breathing, heartbeat, peristalsis. When you are swallowing the food, when food goes in the throat, you can't tell the food that, come back my food. No way, it has gone. The sweetness is gone. Once it reaches the throat, down the stomach. You can't control. You are sleeping as the food is moving. How did it go to the rectum? So that you can go to the toilet to do the best action ever. Yeah. How did it go there? It goes there. You don't have control over that. All those actions are being controlled by medulla oblongata. Hence, the impulse is being sent to medulla oblongata. So what does the medulla oblongata do? The medulla oblongata will send a message, another impulse, to respiratory muscles. That's the diaphragm and the intercostal muscles. Yes. To increase the rate and the depth of breathing. Heartbeat. Meaning that the heartbeat will increase. It does not only increase the heartbeat, but even the depth. The rate, that is fast, the rate, depth. You understand? The depth increases and the rate at which you are breathing also increases. Why does it increase? Why do we need to increase the breathing rate? And the heart is supposed to increase the rate at which it is pumping blood, so that this blood can transport this carbon dioxide from the cells where carbon dioxide is being produced from to respiratory surfaces or respiratory centers, that is the lungs. And more oxygen is taken from the lungs to the muscles or to the cells where this oxygen is needed, so that now the level of carbon dioxide decrease and the level of oxygen increase in the blood. You understand? This thing is working antagonistically, because here we started with carbon dioxide increase and oxygen decrease. So what we are looking at, carbon dioxide to decrease and the oxygen to increase. Therefore, the heartbeat increase. Don't forget that the heartbeat originates from within itself. It has muscles. Yes, tissues. There are muscles, tissues, which initiate the heartbeat. But here, this medulla oblongata, what it does, it increases the rate at which the heart is beating. Yes, when you go to the heart, you will study about it, that it has its beating. The beating is originated within itself. And always I tell my students, we don't determine that the person is dead by using the heart. No. You can remove the heart, hold it, and the person is still alive. What about those people where heart transplant is being done? The hearts are being removed. Yes. So death is not related to the heart. Why? Because the heart has its own beating mechanism. Death is regarded to the brain. If the brain dies, then automatically you are dead. However much the heartbeat is still there, you are dead. Yes. So the heart But this medulla oblongata, why am I saying that? I'm just trying to show you that the function of medulla oblongata is not to start the heartbeat, no, to increase the rate of heartbeat so that more carbon dioxide is removed from the body. And the moment carbon dioxide is removed from the body, definitely the carbon dioxide level is going to fall down. What about the pH? The pH is going to increase back. It's going to go back to normal. Why? Because now there is no carbon dioxide reacting to this, or if it is there, it's very minimal, reacting with water to form carbonic acid. It means that less carbonic acid is being formed. And definitely what happens? The level of carbon dioxide goes back to normal. And that's the mechanism. I don't know why they like this question so much. Maybe because students tended to fail this question, but they like to ask this question. And this is the only question, can I repeat? Is the only question in the negative feedback mechanism where it is brought as it is that describe the negative feedback mechanism of carbon dioxide. The other questions of this homeostasis negative feedback mechanism, they only come in different versions the way I'm going to show you. But this is the only question which comes straight. And in most cases, when it comes for six marks. So guys, just in case it comes, make sure that you go when you know it. It must not be an issue for you to fail. Yes. Let me know if you have any question. Yes, so that I can also smash it before I continue. So let me do a wind up. Number one, stimulus. What is the stimulus? Increase in carbon dioxide level. This leads to the decrease in the pH and decrease the level of oxygen. I explained it, that oxygen decreases because it's being used under respiration and carbon dioxide is being produced, and carbon dioxide reacts with water to form a low pH. And then now the pH is being detected. Because the pH is low, is being detected by the chemoreceptors. Why chemoreceptors? Because chemoreceptors, they detect chemicals, and low pH carbonic acid is a chemical. So now this will, the control centers will send the impulse to medulla oblongata. Why medulla oblongata? We say that it's medulla oblongata because medulla oblongata controls all involuntary actions. The respiratory muscles of the diaphragm and the intercostal muscles, the increase in the rate and the depth of breathing, and muscles of the heart increase. So the moment all this increased breathing increases with the depth and the rate of heartbeat also increases, definitely more carbon dioxide is going to be removed from the body. And then if carbon dioxide is removed from the body, then automatically the pH is going to return to normal. And then it continues like that. I think I'm clear there. And I've said that this is the only question where they bring it as the way it is. You write it the way it is. You don't draw flow diagram. No, please don't draw flow diagrams. We won't give you a mark. Write step 1 in paragraphs. You can write in paragraphs or in bullets. Still is fine. Still you get the mark. Yes. Are we together? Are we together, guys? Yeah, so basically this is an explanation in words of what I've explained. How to write in exam: high concentration of carbon dioxide lead to formation of carbonic acid. The pH of the blood drops, meaning that there is more acidic. It's more acidic. The impulse is sent to medulla oblongata, and the breathing rate and the depth of breathing increase. The heart rate also increase, and more carbon dioxide move to the lungs. Yes, so that it is expelled. Yes, breathe out. All those words can be used concurrently. They're saying that then the blood carbon dioxide level return to normal. Simple. Hey, that's my slogan. I like it because these things are simple, but sometimes the books make them to be difficult. Use this distinction material. There is no way how you can fail to get a distinction. Yeah, download it from the description below. Yes. So it's simple. I always tell my students that it's simple. Things are simple as if you are taking ice cream, because there are no bonds. Anyway, let's look at regulation of water. Yes, we're going to look at regulation of water, regulation of salt, then thermoregulation. Yes. After that, maybe we shall use some 20 minutes and then we discuss about the questions you might find in the paper. If I don't exhaust all of them, definitely I'll come back again only for questions. Only for questions. Yes. So that at least, and I think I should make it either Microsoft or Zoom or website, any of those options. Yeah, meaning that if I don't make it live on YouTube, then automatically I'll make it live the other way. Yes. Okay, so we are done with the carbon dioxide. Let's go to salt. These ones are simple. These ones are simple. Let me show you that how simple they are. Regulation of water of water balance in the internal environment, which we call osmoregulation. Whenever you talk about osmo, we mean whenever you talk about osmo means water. Don't forget that we said osmosis, movement of water molecules from region of low concentration to region of high concentration across a semi-permeable membrane, or movement of water molecules from a region of their high concentration. The water molecules where they are highly concentrated, meaning that it's a dilute solution, to a region of what? They are low concentration, meaning that a high concentrated solution across a semi-permeable membrane. Meaning that whenever I talk about osmo, we are talking about water. So, don't forget the technique I taught you, that if you have decrease, here is decrease, it starts with decrease. By the way, guys, it does not mean that because this thing is facing up, then it must be always increase. No, it is not up. This is not up, and this is not down. It's just a screen. It's just a screen. When you draw, when you do Africa, and you see that the river is flowing like that, have you ever seen a river flowing from low to high? No, there is no river. It's just a paper, or it's just a screen. So this is not necessarily meaning that this is up, this is down. No. So please take note of that. So it must not confuse you that whenever you see something going up, then it is up. No. So now we are saying that there is a decrease. Definitely the last more, it must be increase or rise or more. Anything which is related to increase. You understand? It must be, because we started with the decrease. So now the two steps are done. Now I need to know the gland, which hormone does it produce, and the function of the hormone being produced here. And then I'm done. The negative feedback mechanism or the osmoregulation is done. Simple. Okay. Water level decrease. The pituitary gland. Some students, they have confusion here. What hormone produces ADH? Sorry, which gland produces ADH? The gland is hypothalamus. Why are we talking about pituitary gland here? Okay, let me give you an example, an answer. The hormone ADH, antidiuretic hormone, make sure that you know how to write it. Antidiuretic hormone, yes, is being produced by hypothalamus mass, but is stored in the pituitary gland. So when you use hypothalamus here, make sure that you know how to link it. So is stored because the pituitary gland or hypophysis, sometimes we call it hypophysis. Please don't call it master gland. Please, we won't give you a tick. If I ask you what is my name and you tell me I'm teacher, that's not my name. That's my job. So when you call it master gland because it controls most of the glands. So please don't call it master gland. Call it hypophysis or pituitary gland. Its name is pituitary gland. You understand? Or hypophysis is cool. So secrete more ADH. What is the function of ADH and where does it work? ADH works in the kidney. It works in the kidney where the nephrons are. Yes. So it works in the kidney where in the nephron. It brings about high permeability. Permeable, when you talk about permeability, we are talking about allowing some substances to go through something. Yes. So we call it permeability. Highly permeable. You understand? We call it highly permeable. So it allows, when you talk about high permeability, allows some substances to go through. So ADH, the function of ADH, it goes to the nephron. I think you remember the structure of a nephron. If let me show you here. In the distal convoluted tubule, because this is grade 11 work, but some people now, they have forgotten the structure of a nephron, but it's simple. Let me show you how it looks like. Okay, if this is the Bowman's capsule, it's grade, I'm just drawing a simple thing so that you can understand it better. This is the proximal convoluted tubule. Yes, it goes down like that. Proximal convoluted tubule. Yes, now you have the descending loop of Henry, and then you have the ascending loop of Henry. Then you have yes, you have the distal convoluted tubule. The distal convoluted tubule, and then it's connected to the collecting duct. And here you have the afferent vessel. Here you have the glomerulus. Yes, the glomerulus, and then you have the efferent vessel. Afferent vessel must be bigger than the efferent vessel. That's not a big deal. That's grade 11 one. So what I want is ADH. This ADH will come here in the distal convoluted tubule and the collecting duct. This is the collecting duct which connects to the ureter and then to the bladder. Yes, it comes here and makes it more permeable. Because we have less amount of water in the blood, meaning that you're urinating urine which is yellowish. When you urinate urine which is yellowish, you just know that you have less amount of water in the blood. Therefore, you're supposed to drink more water. Meaning that more water is being reabsorbed back to the blood. Or if you eat something which is salty, if you eat meat, people who go to cheese and nyama, yes, you eat that hot, nyama, yes, please make sure that you drink water. Why? Because these proteins, they require more water for it to be metabolized. Yes. So now what happens is this ADH will make this nephron, the walls of the nephron, this one, the distal convoluted tubule, to become more permeable. When it becomes more permeable, it means that now more blood, more water is being reabsorbed. The word is reabsorbed back to the blood. And then now less is being lost as urine, because what remains here in this tube is urine. So meaning that less water is lost through urine, and then now more water is being reabsorbed back to the nephron, to the bloody vessels. So that's why here we are saying that the pituitary gland will secrete ADH. That one is fine, but this ADH must be more. What is the function of ADH? I explained this so that you understand the idea behind. the permeability of the collecting duct and the distal convoluted tubules will increase. The permeability, meaning that it will become more permeable. It will allow more water to go through. You understand? Yes. So where here in the distal convoluted tubule and the collecting duct, and then if more water is being reabsorbed back to the blood, meaning that more water is the word is guys. Please don't use more is absorbed. We will cross it with the happiness. Why? Why happiness? Because I've told you and you have not taken it as a note. Please don't use absorption. Is reabsorption. Now someone can ask me, why reabsorption? Is because the first absorption occurs in the digestive system. This is the second absorption. Hence we say reabsorption of water to the blood. Can I please have this video after the lesson? Yes, the video will be there. Yes. Guys, don't forget to subscribe, like, and share to others. Ne? Yes. So, we are saying that, I think I've answered you. They're saying that more water is reabsorbed back to blood. And then, once more water is reabsorbed, automatically the water level increase. That's why you say that decrease here. The last statements must be increased. More water is reabsorbed back to the blood. In other words, you're gonna produce more concentrated, concentrated urine. The urine is gonna be more yellowish. Less water is gonna be lost as urine, and more salts are gonna be lost as urine. That's the meaning. What about when the level of water in the blood is too much? We said that our body is made up of over 75% water. I hope we need water. Don't forget that too much of anything is always bad. Meaning that however much water is important in our life, but too much of it is bad. Yes. So, water level increase. Definitely the last statement must be water level will decrease. Or anything which is related to decrease. Less water. More water. I think. So now I need to know which gland. I know the gland. Meaning the pituitary gland. So what's the function of pituitary gland? Is gonna, because I have more water, I don't need more reabsorption here, then automatically I'm gonna produce less ADH, or completely stops. ADH will completely stop depending on how much water is there or is needed in the blood. And then now, meaning that the pituitary gland here, the hypophysis, is gonna stop secreting water. Yes, the water, the permeability of the collecting duct and the distal convoluted tubule will decrease. Meaning that less water is going to be reabsorbed. Meaning that more of it is going to be lost as urine. This is the time when you urinate urine, which is yellowish. Colorless, not yellowish. Colorless, meaning that you have a lot of water in the blood. And this is the time where I don't think you might feel thirsty. You might have that thirstiness. Why? Because you have a lot of water in the blood. This is it. When water level is decreased, you produce yellowish urine. That's the time where you're gonna be thirsty. Yes. And if water is colorless, then that's the time when the water is not going to be, the urine is not going to be thirsty. Did I say thirsty? Colorless. So when it is yellowish, it's smelling too much because of the other waste products which are there. But when it's like this, then automatically, when it's colorless, it's not going to be that you are smelling, because the water has diluted those other ions and wastes. So basically, when there is too much water, the last statement must be decreased. The pituitary gland will stop or produce less ADH, you use 1. You don't write slash, yes. The permeability of the collecting duct and the distal communicating tubule will decrease, and then less water is going to be reabsorbed back to blood. And meaning that more is going to be lost as urine. Guys, am I clear? Are you getting it? Do you have anything you have added to your knowledge today? Yes? Do you have anything? Young Sarah is asking, when is the next class? Friday and Saturday. Yes, Friday and Saturdays. All right. Let's continue and say that when blood has more water, okay, this is just a description of what we have explained. When a blood has less water than the normal, the hypothalamus is stimulated. You see, I told you that you need to know how to connect if you are using the word hypothalamus or you are using the pituitary gland. Both of them are fine as long as you explain them correctly. Then the hypothalamus is going to be stimulated and is going to send the impulse to the pituitary gland. Why it sends the impulse to the pituitary gland? Because the pituitary gland stores it. And then now the pituitary gland is going to secrete more ADH. You see? Then this ADH is going to travel in the blood to the kidney. Where exactly in the kidney? In the nephron. And then this ADH is going to increase. What's the function of that hormone? Is to increase the permeability of the nephron. And then after that, water is reabsorbed back to the blood. Meaning that less is lost as what? As the urine. So water level in the blood returns back to normal. And then the other way is the same thing. When blood has more water than the normal, the hypothalamus is stimulated and then the impulse is sent to the pituitary gland. And then what happens? Less ADH or no ADH is produced. So you choose whatever you want to write, either less or no, or depending on the scenario they have brought you in the exam. So less travel in the blood to the kidney. And then what happens? There is decrease in the permeability, meaning that the walls, they're going to become less permeable. It does not allow water to go through. And then now less water is reabsorbed back to blood, meaning that more is lost as what? As urine. Here less is lost as urine. Here more is lost as urine. This is yellowish urine, and then this is the colorless urine. Yes. And then now this is smelling urine, and this is the, I don't know, odorless. Water level in the blood returns back to normal. I think the way how I've explained it here. What about the salts? So you guys, we are remaining with the salts and thermoregulation. I'll show you how you can go to the paper without stress and you pass everything. Why not passing? You have to. Yes. Because they set these things so that you guys, you can pass. So you guys make sure that you pass. Make sure that you pass. And here they're saying that regulation of salt balance. How to write in exam. But before you know how to write in exam, you need to know how to work upon it. So it's the same thing, guys. It's the same, exactly the same thing. If you are saying that normal level, cool. Increase here, decrease in the level of salt, yes. The last statement will be increase in the level of salt. We know that. Then you need to know the gland which is produced, which is going to act upon the salt. is adrenal gland. Don't forget that adrenal gland produces two hormones. The first hormone is aldosterone. The second one is adrenaline. So in this case, we don't want adrenaline because adrenaline is used as emergency. It's a fight or flight. You either take off, or you have to fight. So that is adrenaline. We're not interested in adrenaline now. We're interested in aldosterone. So aldosterone is going to be produced because we have less. So what's the function of aldosterone? It also brings about the permeability. It brings about more reabsorption. I said that always use the word reabsorption. So adrenal gland, what's happening with adrenal gland? Adrenal gland is going to produce more aldosterone, and this aldosterone is going to make the nephron to reabsorb more sodium ions. The moment sodium ions are reabsorbed, the salt level will increase and goes back to normal. What about when the salt levels are increased in the blood? Then the last statement must be decreased. So adrenal gland will stop or will secrete less aldosterone, one of the two. And then now it means that it will be less reabsorption of sodium ions, or there is decrease. The sodium ions reabsorption will decrease, and the moment decrease, the level of salts will go back to normal. Now, this salt, aldosterone, aldo, which means salt, salt, aldo, just for you to remember. It's just for you to remember. It's not a big deal. Yes, this aldosterone, works together with ADH. If there is too much salt, sodium ions, too much salt, yes, meaning that there will be less reabsorption of salt. It means that there is less water. Too much salt, it means less water. Too much water, if there is too much water, then it means that there will be less salt. So these two, they work antagonistic in the opposite way. Yes. So meaning that when ADH brings about more reabsorption, there is a question I saw. I'm gonna give it to you so that I show you how you can answer, how you can bring about the interaction of aldosterone and the ADH at the same time. So that you must know that when too much water is there, you urinate urine which is colorless, it means that that urine has less salts but too much water. When you urinate urine which is yellowish, it means that that urine has more salts but less water. And that's exactly what happens in the body. Yes. So I think I'm clear there. Then how do we write in exam? How do we write in exam? We write in exam that when the salt level in the blood increases, yes, the adrenal gland will be stimulated. When it's stimulated, what happens? It's gonna secrete less. If it is less, it's gonna produce less. It's gonna produce what? Less aldosterone. If less aldosterone is there, it means that there's gonna be less reabsorption because there is too much salt. Yes, less reabsorption, and if less reabsorption occurs, what happens? It means that there will be a decrease. There will be a decrease in the salt concentration in the blood, and then to turn back to normal. What about when there is increase in the salt? So there is decrease in the salt. So the last statement must be increase, as I told you, meaning that the adrenal gland will become more stimulated, meaning that more aldosterone is going to be produced. And if more produced, meaning that there will be more reabsorption. That's why you're seeing that increase in the reabsorption of sodium ions. And then once more sodium ions are reabsorbed back to the blood, meaning that the level of salts in the blood will increase and it will turn back to normal. So you see that things are not as that difficult as you see them. The only challenge which happens is when it comes to the exam, sometimes, my students, you go off. So after covering this, then it means that I'm going to be focusing more on these topics. You understand? So the next topic after this, a link so that at least you don't get lost in that regard. Let's go to the last subsection. What is that? Is the process of thermoregulation. The process of thermoregulation. Here, when you talk about thermoregulation, what comes into your mind? Thermo means heat, temperature, thermometer. Whenever I talk about thermo, we have to talk about heat, temperature. Regulation is control. Basically, is you are controlling the heat, the amount of heat in the body. So when you talk about thermoregulation, is controlling the amount of the heat in the body. Remember that we are endotherms. We're supposed to keep our internal environment constant. That's why thermoregulation is also an example of homeostasis. It's supposed to be kept constant. Above it is problem. Below it is problem. Meaning that we're supposed to regulate at 35.5 degrees Celsius. This is 5, not 8. Ne? Yeah, 35.5 degrees Celsius. Yes, at least there. 35 to 30. Yeah. Did I say 35? 37. 37.5 degrees Celsius. So above that, yes, above, not 30, 35, ne? 37. I was talking about the temperature at which sperms are being produced. Anyway, it's 37.5 degrees Celsius. So above it is problem. Below it is what? A problem. Meaning that when it's above it, it will be denaturation of enzymes. And then you won't work. You won't have hormones. You won't have enzymes. Because all those things are protein in nature. Anything which is protein in nature will be denatured. And then below it, it will become inactive. Inactive, meaning that it will be slow. It will be dormant. Yes. Like the same way some creatures are dormant in winter because the temperatures are low. What happens when it's too hot? You go to the houses also. Meaning that too much of the temperature is bad. Too low of temperature is bad. Anything which is too much is always bad. Anyway, let's go back to what brought us here. So, we're saying that thermoregulation is the process that allows your body to maintain its core internal temperature. All thermoregulation mechanisms are designated or designed to return your body temperature to a steady one. So the body temperature is regulated by hypothalamus in the brain. Meaning that the hypothalamus is supposed to communicate to the blood vessels and the sweat gland. Meaning that during this process, we're going to talk about more of hypothalamus, the sweat gland, and the blood vessels of the skin. So once you know that, that's when you talk about the thermoregulation, you must know that that's what you are supposed to talk about. Please don't forget this, because this is your future. Okay, before you go there, you need to know something very small. If I bring a diagram for you, can you identify for me the following? Number one, the things which you need to know is sweat gland, sweat duct, sweat pore, sweat. Yes. Sweat gland, sweat duct, sweat pore, sweat. Then the blood capillaries. So when you talk about sweat gland, yes, when you talk about sweat gland, we talk about this. Sweat gland. Yes. This is the sweat gland we are talking about. Sweat gland. Sweat duct, a tube which transports the sweat. Sweat duct. Sweat duct is this one. And then sweat pore is the hole where sweat moves out. Is this one, sweat pore, where you see your liquid when you are too vigorous with an activity. Then the liquid which comes out. So you have to know how to label it. Then number two, you need to know the blood capillaries. The blood capillaries. This is a hair follicle. So the blood capillaries, you must know them, how they work. Yes. So now let's come here. So this is the blood capillaries, and this is the sweat I was talking about. So sweat gland, sweat duct, it's a sweat duct. Then this is a sweat pore, and then this is sweat. So you need to know that. And then these are the blood vessels, blood capillaries. So we have this shunt. During hot day, it shrinks and more blood will pass through near the skin. And then during cold day, then this opens and then this shrinks, and then more blood, this becomes bigger. Let me show you. It becomes bigger, you see, and then this one shrinks, and then more blood will be passing down the skin. And that's why during hot day, you cannot see, during cold day, you cannot see blood vessels on your skin easily. You can see, but you cannot see them easily the way how it is hot. Yes. So now, what happens during this thermoregulation? What happens? Let's link now. Let's link to the question. All right. Let's go back to the question, and then we see what happens on hot day. So now we have to discuss about how do we bring questions concerning about this. Yes. Anyway, let's go. The first statement you need to talk about is the hypothalamus, because it is the hypothalamus which detects the change in the temperature. Don't forget that we said that the function of hypothalamus, when you go back here to this, the hypothalamus, yes, we say that is responsible for the hormone it produces is ADH. But here we say that yes, we talked about it when you're talking about the hormones. Down here on this booklet, let me show you, just to remind you something small, here. Guys, make sure that you have this distinction material so that you can, yes, hypothalamus, where are you? Say, I'm here. Okay, it's here. All right. Control center for hunger, thirsty, sleep, body temperature. That's why you talk about hypothalamus, because hypothalamus is responsible for body temperature and emotions. Are you always happy, sad, bipolar? Meaning that now you're happy, next time you are sad. What controls that is hypothalamus. Anyway, this is not today's subtopic. Today's subtopic is just to know why do we talk about a hypothalamus? Because we said here, hypothalamus is the center for temperature. Now, let's go back to our description. Yes. So, now when you're saying that, hypothalamus is stimulated. Now, I think you know the reason why it's stimulated. During hot day, when you talk about hot day, hypothalamus is stimulated. When it is stimulated, what happens next? Yes, it sends the impulse to the blood vessels. Meaning that these blood vessels, this hypothalamus is going to send the impulse to these blood vessels. What will happen to the blood vessels? The blood vessels will dilate. They will become big. The blood vessels of the skin, meaning that they will become more wider. This is called vasodilation. Guys, listen carefully. You're saying that hypothalamus is stimulated and sends the impulse to the blood vessels. That's the mark. The blood vessels will dilate. That's the mark. This is called vasodilation. Is another mark. So don't write the blood vessels will become dilated and you skip. You go to more blood, you will float near the skin. No, this is called vasodilation. We give you a tick for that. This is called vasodilation. More blood flows to the skin, meaning that we will experience more blood flowing near the skin. Yes. And then more hot. Yes. Here you have more heat is being lost from the skin. If more blood is flowing near the skin, meaning that we're going to have a lot of heat being lost. That's why you see these arrows there. Why? Because this blood is hot. So if it's hot, meaning that more blood is going to be what? It's going to be lost. More blood is going to be what? It's going to be lost. And then, if more blood is being lost, what happens? It means that, more heat is going to be lost. What happens? Meaning that it's going to make the blood to be cooled a bit. But it does not stop there. More blood is sent to the sweat gland, meaning that now this blood is going to go to the sweat gland. More blood is going to be sent to the sweat gland. Now, the moment more blood is sent to the sweat gland, what happens when you put too much food in the mouth? You just stop chewing, or you increase the rate at which you are chewing. You increase. So if more blood is sent to the sweat gland, it means that the sweat gland will become more active. The sweat gland is going to become more active. And once it becomes more active, it means that more sweat now will build. You understand? And then now you'll see your skin like this. Meaning that more sweat is being produced, is being released. So the sweat gland will become more active, meaning that it's going to produce more sweat. More sweat is being released. And once it releases, is being released, it means that now, as it's being released, evaporation is going to take place. And evaporation to take place, you require heat to evaporate this water. And as evaporation is taking place, it leads the skin to be cooled. It leads to the cooling of the body. So evaporation of the sweat cools the skin. That's the meaning. Yes, that's the meaning. So that evaporation leads to the cooling of the skin. Sebastian, brilliant. Okay, thank you very much. Yeah, actually, you are more brilliant, the one who is listening. Yeah, than me who is talking. Because when you are taking the information into the head. Guys, let me know if you have a question, so that we sort that out. If you have any challenge, don't be scared to ask a question. Don't be scared. Ask it, we smash it. So that at least you don't lose it. Yeah, you don't lose it. So basically, that's what happens during hot day. What happens during cold day? What happens? So the hypothalamus, because it is the same gland, is going to be stimulated. But in this case, when it's stimulated, it has to send something. The impulse sent must be just a little different. Okay, the impulse is going to be sent to the blood vessels. The blood vessels will constrict. Guys, please, those who are having the distinction material need to rectify this. Yes. The blood vessels become wider. No. The blood vessels will become narrow or small. Yes. They'll become narrower. Yes. This is called vasoconstriction. The blood vessel of the skin will become narrow. This is called vasoconstriction. Check. Check. The upper side is narrower. The lower side is big, wider. This is a tick, a tick, a tick. Yes. Guys, did you know that it is nice to mark a student when you are just giving tick, 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 tick? Make the hand of the examiner just to tick, so that even if he's wrong, they just give you tick, 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 tick. Then when you get wrong, the examiner starts to say wrong, even the correct one wrong. Aibo. Make the examiner to be impressed that everything you are writing is correct. Ne? Yeah, how are you going to make the examiner? Use this distinction material. Download it, guys, and get your copy. Watch these videos. Funded Duke. Yeah, you always. There is no way. I remember this guy who WhatsApped me and they said that, sir, I got 12 distinctions from my class. He was a teacher. They said, this is brilliant. 12 distinction. He had never got a distinction before. But when he started using this distinction material, distinctions now. Distinctions are just flowing like water in the river. Flowing, water in the river, water in the river, water in the river. Yeah, so make sure that use this distinction material. You will have distinctions flowing like water flowing in the what? In the river. Yes. So we're saying that if this is vasoconstriction, less blood will flow near the skin. We have explained this. Less heat is gonna be lost because less blood is being is flowing near the skin. It's like you when you are sleeping without a blanket, more heat is lost. What about when you have a blanket? Meaning that the heat will take much more time to cross this blanket to go out. Meaning that less heat is gonna be lost. Yes. So less blood is sent to the sweat gland. If less blood is sent to the sweat gland, meaning that the sweat gland is going to become less active. What happens if you put less food in the mouth? Definitely, you'll chew less. That's the same thing. Meaning that here, it's not that much, meaning that you see less sweat is being produced. Meaning that you won't have that much sweat. Like in winter. Yes, less sweat is released and then less evaporation. And if less evaporation is happening, it means that there will be less cooling of the body. Then you'll be like this. Why do you have these goosebumps? And the hair are going up. Anyway, this hair rising is also another mechanism of keeping internal environment constant. Unfortunately, we don't talk about it in this case. But I would have explained it to you. If you go to the university, they will explain it better when you are using this erector pili muscle, which contract and make this hair to stand upright. Yes, anyway, you're not talking about it. Let me not put you in the stress. Wow. Here are some of the questions. Yes, here are some of the questions which you need to answer for me. Can I give you 2 minutes to answer for me how to explain the negative feedback mechanism of carbon dioxide when a person is running faster? I explained it, young sir. I explained it. It's simple thing. The way. That's why I told you that the carbon dioxide. Wait, guys who are starting to write. The carbon dioxide is the same. Is the same. You don't need to have stress with it. That's exactly what you need to write. Yes. So meaning that when the person is running faster, it means that more carbon dioxide, more oxygen is being used, meaning that respiration is taking place, meaning that more carbon dioxide is being produced. Meaning if there is more carbon dioxide being produced, meaning that there will be more carbonic acid which is being formed, and the pH of the blood will drop. You understand? And then the impulse is going to be sent to the medulla oblongata, and then the breathing rate and the depth of the breathing is going to increase. You understand? And it doesn't stop there. The heart rate also increases. And then now more carbon dioxide will be sent to the lungs through the blood. And then the lungs is going to exhale this carbon dioxide from the blood. And then the level of carbon dioxide will return back to normal. And once it returns back to normal, then the person will go back to normal in terms of the heartbeat and the breathing. Exactly. I said that negative feedback mechanism for carbon dioxide is the only mechanism where whatever case they bring it, you're going to have to write that. You understand? Yes. You're going to have to write that. And then it doesn't have the other way around, meaning that there is no way how you're going to say that, okay, the top side or the bottom side. Which one am I going to choose? You only choose one. That is it. So carbon dioxide, whatever case they bring it, you write the same thing. Make sure that you memorize it and then you get it right. I think, young Seri, I think I've tried to explain that. All right, let's go through this. And then we say that. Identify the part labeled A. Where is A? A, yes, we have said it, that it is sweat gland. Yes. Give the number of the diagram 1, 2, 3. 1. Oh. Give the number of diagram 1, 2, 3 that represent the body's response to high environmental temperatures. Here we have to look at what do I look at if the temperature is high? If the temperature is high, yes, if the temperature is high, you need to look at the blood vessels. They must be dilated, and you need to look at the sweat being produced. You can't look at the sweat gland, because the sweat gland, you don't know whether it's more active or not active. The only thing you know that is active or not active by looking at the amount of sweat which is being produced. So on the hot day, it's going to be this and this, because here they check the blood vessels. These ones, they are more wider compared to this. You see, they are more wider. Check this size. They are more wider compared to this. They're saying that would the skin release more heat through radiation? Diagram 1 on diagram 3. Remember, radiation is a loss of heat without contact, meaning that like the sun, the way how it brings heat to us, you don't touch the sun. Yes. So they're saying that would the skin release more heat through radiation in diagram 1 or 3? Diagram 1 or 3. Because this one is under high temperature, then it's going to lose it diagram 3. Give number of the diagram 1, 2, 3 that represent vasoconstriction. 1, 2, 3. Vasoconstriction is diagram 1. Why? Because the blood vessels are constricted, meaning that they are small, they are narrow. They're saying that which part of the brain controls thermoregulation? Hypothalamus. We have talked about it, and then even I showed you about it. What about this question? They're saying the diagram below represents the structure of the skin. Structure of the skin. They are saying that if this is the structure of the skin of two people, both people were in the same room at the same time, but one person was exercising while another one was not exercising, meaning that he was sitting still. The skin surface temperature of both people was measured after 10 minutes. Okay, which person A or B was exercising? Let's look at what we said. Here there is sweat, and the blood vessels here are more wider, meaning there is vasodilation. So it's this one who was exercising. Give two visible reasons: production of sweat, and then the blood vessels are more wider. Give one hormone that would have the same effect on the blood vessels that is observed in A. In A, there's vasoconstriction. You see? If it is vasoconstriction, then definitely the hormone is going to be adrenaline. Why? Because during emergence, we said that the blood vessels of the skin constrict. Let me show you. This distinction material has this answer. As I told you that there is no way how you're gonna be asked a question and the distinction material, this book cannot answer it. So let me show you where it is. You go to where am I here? Where am I? Yes. So it's around here. Yes. Let me link where. What is this? Okay. Okay. Now it's around here. Sympathetic and parasympathetic. Yes. Check. We say that sympathetic nervous system work together with adrenaline. You see? And then what happens? During the sympathetic nervous system, that's when adrenaline works together with the sympathetic nervous system to bring about emergence. There will be increase in the heart rate. Cool. Constriction of blood vessels in the skin, vasoconstriction. You see? Definitely, this is what they are trying to say in that question. Wabon. So, if we go back to add that question, yes, if you go back to this question, here, they're saying that name one hormone that would have the same effect as blood vessels that is observable in the person 1. This is vasoconstriction, and so the hormone is gonna be adrenaline. Yes. After 10 minutes, the surface skin temperature of each person was measured. Okay, if the surface skin temperature was measured, so what does it mean? It means that the person A, so explain why the skin temperature of person A was higher after 10 minutes. And person A was the one who was exercising. You see? So why is nope. Explain why the skin temperature of person let's find out. Did they say person A is the one who was exercising? They're saying after 10 minutes of the skin surface, okay, the person was measured. The result is here. So they're saying that explain why the skin temperature of person A was higher after. Where is person A? We need to find out. Person A. They're saying the diagram below represents both people, okay, exercising, and another person was sitting still. The skin surface temperature of the was measured after 10 minutes, meaning that this one was exercising while this one was not exercising, because of what we are seeing here, because of the heat, and even we said it, I think somewhere here, who was exercising, and we say that person B was the one who was exercising, meaning that person A, his temperature was higher. Person A, temperature was higher, but this person was not exercising. While person B, the skin temperature was lower, and this person was exercising. But this only happens after 10 minutes. What caused this? It's because of this. It's because of this. Meaning that more heat was lost to the surrounding. The moment more heat was lost, the surrounding was being absorbed by, and then now this liquid, the heat, the sweat, also led to the cooling. And then the moment it cools, so when the air blows, on this one becomes colder. You understand? And then now the skin surface, it becomes much more colder. It's like when you take a warm bath. Yes, you bath. When you bath, it is you are warm. But when you remove out of the warm bath, yes, and then you expose your skin to cold air, you become more colder than before. That's exactly what happens here. That this person, because the person A was not exercising, so was not exposed to any coldness, any liquid which is going to make the skin to be more cold. But now because this one was exercising, and then the sweat was there, and then the sweat made the skin surface to lose the heat, then the skin surface became more colder. That's why this person is having a lesser temperature compared to this, but only at the skin. There is another question. There are so many questions, guys. Yes. Here is a question. They're saying that normal healthy person was placed on. Okay. If there is any question, please let me know. Yes. They are saying that a normal healthy person was placed in a cold room after 30 minutes. The thermal image shown below was placed. Okay. This thermal image indicates the temperature of different parts of the body. Lighter colors of the scan indicates the temperature lower than the normal temperature. Oh, okay. Meaning that this is lower than this is lower than this. Which color, gray, black, or white, represents the body temperature? Definitely is black. Why? Because they say these are lower than. So this is the normal body temperature. So the answer becomes black. State what occurred in each of the following parts in the person's skin during the temperature regulation. Okay. Blood vessels. Now you need to find out, was it in a hot environment or in a cold environment? Cold room. Definitely you're gonna have, if it is cold room, then automatically blood vessels will go constrict. Yes, this is what I'm saying. They will constrict. This is what I'm saying. Because it is on a cold day, constrict, vasoconstriction. They'll become more narrower. And then sweat gland. The sweat gland will become less active. The sweat gland will become less active. That's how so it's going to become less active. And they're saying, name the part of the brain that is responsible for thermal regulation. Hypothalamus. We have said it several times. Hypothalamus. Yes. There is no way, guys, if you follow this distinction material, you're going to fail. Name the part of the brain, okay? State one way in which the thermal image would differ if the person was placed in the hot room for 30 minutes. So how will it differ? Yes, how will this? Definitely, automatically, it means that these light colors will become much more darker because the heat is being absorbed from the surrounding. Yes. So basically, guys, there are a lot of questions you can talk about, but I think for today, should I stop here? Or I should give you one more question. Let me see it here. www.thundereduke.com thundereduke.com. Just to pick another question paper for you. thundereduke.com. All right. Yes. Okay. Okay. So thundereduke.com. Okay. Grade 12. All right. Grade 12. Yes, just to show you another question maybe, so that at least grade 12 study notes here. Yes, life science grade 12. All right, life science grade 12. And then yes, let's look at let me choose which year. Let me check this, see if it can work for you. Is there any question? Definitely the question is there. Okay, oh, that is the same question. It was that year. Okay, let me then use this 2019. Okay, let me share this with you. Okay, open. All right, I think now you can see it. You can see it. Let's look for the question here. Definitely there will be a question. I don't know which question is it, but there will be a question. A hormone that stimulates mammary gland, that's prolactin. No, I'm just passing, and then I saw something nice of what we have tried to cover. Okay, definitely there will be a good job. There is a question here. Is there any other question? What is this? Yeah, diabetes mellitus. Yeah, there's another question. Okay, now let's go through this, and then we wind up our discussion today. And as I said that, our next discussion is going to be the discussion today. So yes. Can we continue, guys? Yes, young Siri, are you there? All right. Let's go to that question, and then we see what are they talking about. The diagram below shows reabsorption of salts. Oh, when they talk about reabsorption of salts, what happens into your mind? Aldosterone and water through tubules of nephron in the kidney. Salt and this question resembles that what I talked about. That salts, remember I told you that salts and salts. You see, I said that aldosterone, the salts and the water, they work together antagonistically. Yes. So that they're saying that the width of the arrow represents the amount of salts and water. All right. Let's start. Reabsorption of these so what is it? Reabsorption. We need a key. Oh wow, the key is here. Dark means salt, gray means water, meaning that this is water, this is salt. This is salt, this is water. Glomerular filtrate. When you talk about glomerular filtrate, I think you know. I drew the nephron. You remember I drew the nephron here. This is the glomerulus. The filtrate is here. What you know when you are sieving, yes, the glomerulus is like a sieve. It sieves, only small particles are going through. So the liquid which goes down is what called the glomerular filtrate. So that's the meaning here. Let me close this so that. And then now reabsorption, meaning that it goes back to blood. This one goes back to blood. And then renal, this goes back to urine. So this is urine, this goes back to blood. This is the glomerular filtrate. All right, diagram 2, diagram 3. Anyway, maybe we shall understand it better when we ask questions. Name the hormone in the human body that is responsible for controlling the amount of water. It's ADH. ADH, and then salt, aldosterone. Name the gland that secretes the hormone above. Which gland is adrenal? Adrenal gland. Yes, adrenal gland. Guys, my handwriting is bad. Yeah, it's too bad. Ne? Anyway, adrenal gland. So when you talk about yes, adrenal gland. Then they're saying that which diagram 1, 2, 3 would represent the person who had eaten salty chips on a hot day, meaning that first of all, too much sodium ions. Aldosterone is working too much, meaning that this person has too much salts in the blood. Hot, meaning that the person is sweating too much. It means that you have less water. Too much salt, meaning that if I have too much salt, so I expect to lose much in the urine. You understand? So if I have too much salt, it means that much salt is going to be lost in the urine. And if I have less water, this is less water because you sweat too much, meaning that you lose a lot of water. So meaning that I have the water, what I expect the water to be, I'm going to be reabsorb more water and lose less water in the urine. So when I go to the diagram, I'm going to look for more salts lost, less reabsorbed. Let's go. More salt. We say the salt is dark. More salt lost. Hey, more salt lost. All right, let's continue. So we are saying that is the process of maintaining a constant internal environment. The internal environment here, it means it can be blood, it can be sugars, it can be salts. So all this water, it can be all the things which we keep which we have inside our body, it must be kept constant. So if you keep them constant, basically, when we say constant, we don't mean that it's supposed to be 9, for example, at level of 9. It has a specific range. As I say, that maybe from 7 to 9, that is normal. So above 9, then it becomes abnormal. Above or less than 7, it becomes abnormal. So this homeostasis, which works together with the negative feedback mechanism, yes, brings it back to this range and brings this one back to this range. So some students, they confuse what you call the homeostasis and negative feedback mechanism. The mechanism of bringing it back to normal is what we call the negative feedback mechanism. Why do we call it negative? Because we have to subtract it. So if it is positive, there is also positive feedback mechanism, but we don't talk about it here. When you go to the universities, you talk about it. But now here, when you talk about this negative feedback mechanism, it means that something which went wrong must be brought back to by subtracting what that made it wrong, then you bring it back to normal. That's why we call it negative. But if something gets wrong and then it becomes more wrong, then it becomes positive feedback. But we don't talk about it. Yes. So here, this is just to bring about the normal functioning of the body so that everything works to the way it's supposed to work. So this enables the body to function efficiently. So when it works efficiently, there is a difference between efficient and effective. So don't use effectively, use the word efficiently, because when you talk about efficiently, yes, the body is supposed to work. When you say efficiently, sometimes the efficiency might not be about 100%. When you go to physical science, you know that better. But despite the changes which might happen in internal and external environment. When you talk about external environment, we're talking about the surrounding areas, what you have, maybe the temperatures of the surrounding, maybe you talk about the humidity, that's the surrounding. When you talk about internal environment, we are talking about maybe the blood glucose, the salts, the water. Did you know that a human being has the same temperature, however much this person goes to water or to the ice, the temperature remains the same? So that's what you're saying. Despite all the changes which might happen, you understand? Yes, the internal environment must be kept constant. So now, if it fails to be kept constant, then now the problem will come in. How does the problem come in? Maybe the system will fail to work properly. I think I'm clear there. Good day, sir. Hope you are well. Thank you very much for simplifying lessons, making it easier for us. Okay, thank you very much, guys. Yes. Make sure that you do what? You share to others also so that they can benefit. Guys, if you have a problem, this lesson is live. This lesson is what? Is live. So if you have any problem, let me know. I can try to answer those questions. I'll be giving you some questions. This lesson might go for 1.5 hours, if not 2 hours, so that we maximize all the things which you might make this situation to be difficult for you, so that you guys will get distinctions, because that's what we are looking forward for. All right, we are saying that changes in temperatures, glucose levels, carbon dioxide, and water levels, plus salts, these are the internal environments we are talking about, which are supposed to be kept constant. Yes, so those are the internal environment we are talking about. So this internal environment must be kept constant. Yes, so we are going to look at them in detail. So they are saying that regulation of glucose levels. Now, here you need to understand some few things before you go to the regulation. Let me give you a trick so that you can understand this better if they bring it in terms of this diagram you see. Let's start with the glucose. Number one, glucose. Guys, you need to know two things from two things. The first thing you need to know is always in the middle here, you have what called the normal. Guys, all of the mechanisms, they look like this. You don't need to stress. So always it is like this, normal. You understand? When you say normal, okay, what is going to happen here? The last statement and the first statement must be in opposite way. If you see here there is increase, yes, the last statement must be decrease. If you see here there is decrease, yes, the last statement here must be increase. I think I'm clear there. Whatever negative feedback mechanism you're gonna talk about, except one, that is carbon dioxide. I'll explain that better regarding to why carbon dioxide is not made to be like that. It's because its mechanism is one way. It's not two ways like these ones. So now we are saying that the first one must be, if it is increase, the last one must be decrease. If the first one is decrease, the last one must be increase. So now you only have to fill in 2 points here. So make sure that you separate these two from these two. So how do I know these 2 points? You need to know what are they talking about. This is also being spoken about here. The normal level of glucose in the blood. So if glucose level increase, the last statement must be glucose levels will decrease. If the glucose level decrease here, the last statement here must be the glucose levels increase. I think I'm clear there. Now, when it comes to glucose, then you have to ask yourself, when it comes to glucose, which gland is responsible for glucose? And what is the function of that gland? So you mean that if you're talking about glucose, glucose goes with glycogen. So glucose, yes, is being acted upon by which hormone? And which gland does it which gland produce that hormone? So it means that the hormone and its function will be here, and the gland and its function will be here. Meaning that the function of the gland is the hormone it produces, and then the function of that hormone will bring about the decrease in that level of whatever you are talking about here. For example, say that glucose level will increase, or definitely the last statement will say that the glucose levels will decrease. So that one is sorted. 2 marks gone. Oh, you have got them before even you go to the paper. Then you have here pancreas, because the glucose, what works on glucose and glycogen is pancreas. So the pancreas, that's the gland, will produce or will secrete insulin, meaning that you have talked about the gland and its function, meaning that the gland, which is pancreas, what is its function? It secretes insulin. Done. Then you come here and you tell us the function of insulin. This insulin, what does it do? You tell us that now insulin will stimulate the conversion of glucose to glycogen because glucose is too much. So insulin will stimulate the conversion of glucose to glycogen. My students, please don't say that insulin will convert glucose to glycogen. We'll get it wrong, because insulin does not convert glucose to glycogen, but insulin stimulates the conversion of glucose to glycogen. What converts this is the liver, because when you go to the university or other areas, you'll find out that we'll include here a liver. Yes, we'll include isolate of Langerhans. When you are specific to then you talk about the beta cells, the alpha cells. We will talk about those things when you go there. But for now, what is most important is, you know that insulin produced from the pancreas, then that insulin is going to stimulate the conversion of glucose into glycogen. Here, that's why here I wrote that glucose is converted to glycogen. What converts it is the liver. What stimulates the liver to convert glucose to glycogen is insulin. So basically, you just know the gland, and here, which hormone does it produce? And then here you come here, you talk about the function of that, hormone. That is the stimulation. So basically, that's what you need to know about it. So when you go here, it's the reverse. It's the reverse. So you don't need to get stuck when you reach there. You understand? Does offer lessons or videos? Yes, the lessons are there. The lessons are there, but unfortunately, I'm not a math teacher. I'm a life science and physical science teacher. So it means that I only offer here life science and physical science lessons, but maths, other subjects, all other subjects are here, but by different teachers. Yes. So if you have questions, still you can drop them in the comments. Any video you comment on, still we will bring your response, or we will try to act on your question you have answered, you have asked. Anyway, let's continue. Then let's go to what about when there is a decrease? If there is a decrease, yes, it means that glucose level has decreased. The last statement must be glucose level will increase. When glucose level decreases, talk about the gland. So which gland is the same? Pancreas. Pancreas is going to secrete glucagon. Don't forget that you have glucose here, glucose, and then you have glycogen. So now here you have, if you want to convert glucose to glycogen, use insulin. And then if you want to convert glycogen back to glucose, you use glucagon. Yes. So now it means that now the pancreas will secrete, because now we want more glucose because the glucose levels are low. So the pancreas is going to secrete, yes, glucagon, and then the glucagon will stimulate the liver to convert glycogen back to glucose, and then you go back to normal. Guys, this is the same situation when you have, for example, you are hungry. It's break time, you are hungry, and then after that, they don't tell you to go to maybe the teacher say that you guys, you have to complete this. Then after a certain time, the hunger will go. But how did that hunger go? It goes because now the glycogen has been converted back to glucose, and then now the body will receive this glucose and it will become normal, or it will become okay again. And then life continues. Yes. So basically, the storage form of glucose is glycogen. The storage form. In animals, the storage form of glucose, if they ask you that question, what is the storage form of glucose, is glycogen. The usable form of glycogen is glucose. The usable form of starch that is in plants is glucose still. So you have to know that, just so that if they bring the question, you are able to answer such questions. Now, this is the same thing I've explained. You don't need to memorize, like, no, cram. You just need to understand. First step, increase. Last step, decrease. Which gland? Okay, you know the gland. Which hormone produces? You talked about the hormone. So what is the function of that hormone? So therefore, you have got to know the function of that hormone, and then after that, that hormone is going to bring about a decrease in the level of glucose, and then the situation goes back to normal. Glucose level in the blood increase, yes, above the normal level. The pancreas is situated in the, is stimulated to secrete insulin into the blood. So what is the function of you see, it goes above normal. You talk about the gland. What does the function of, what is that gland do? It produces insulin. What is the function of the insulin? This one stimulates, you see, stimulates the liver to convert excess glucose to glycogen. You see, once excess glucose has been converted into glycogen, definitely the level of glucose will decrease and goes back to normal. That's what I was trying to explain here, but here is just in words. And then here, glucose level, yes, decrease below the normal. So what happens? The pancreas, you talk about the gland. What's the function of the gland? Produces glucagon. What does glucagon do? The glucagon will stimulate the conversion of the stored glycogen back to glucose. The moment glucose is available in the blood, definitely glucose levels in the blood will increase, and then it goes back to normal. Simple. Simple. Guys, let me know. Is this thing simple or is that difficult? Ne? I don't think these things are difficult. You just need to put love. Some people, they call it love. Yes, put love there. Yes, the way you want it to call it. And then after that, you will see that these topics are very simple. Yes, let's look at some of the abnormalities or the deficiencies here. So I've explained this. I've explained that. And then, yes, Young Seri is saying that you make these topics easier for me. Thank you very much, Young Seri. And I always see you whenever I do my lives. You are always there. So I credit you for that. Ne? Thank you very much. Yes. So let's continue, guys, to the next question. Yes. All right. Let's go to the next section. So here you have glucose. You have glucose. Here you have glycogen. From glucose to glycogen, you use insulin. And then from glycogen to glucose, you use glucagon. Simple. So what produces this is pancreas. And then once you know, guys, go to the paper when you know this. Ne? And then number 2, you know how to manipulate this. Simple. Once the moment you know how to manipulate this, then you will be able to know all this. So we're going to see some of the questions which are concerning about this. You'll see that the questions are simple to answer. Diabetes. Some of the conditions which you find in this glucose situations is diabetes. If the body cannot produce sufficient insulin, if the body, the pancreas, cannot produce sufficient insulin, you're going to suffer from what called diabetes. My students, please don't say diabetes. Because when you say diabetes, we will cross it. If the pancreas fails to produce sufficient insulin, or yes, insulin, then you're going to suffer from diabetes mellitus, not diabetes. Because when you say diabetes, we want to know which type of diabetes are you trying to mean? Because we have 2 types of diabetes. We have diabetes mellitus and diabetes insipidus. The diabetes of sugar and diabetes of water. So now, here, diabetes of sugar, diabetes mellitus, is divided into diabetes type 1 and diabetes type 2. Please don't say sugar diabetes. No, say diabetes mellitus. Simple. Don't complex your life. You bring in, what if I say this? What is the problem for you to memorize diabetes mellitus? Why do you want that? Yeah? Why do you want that? This is what we want. Don't complex your life by losing a mark. Just to say simple, diabetes mellitus. Simple. You understand? Yes, you go to the paper, come out when you're happy. Don't come out of the paper when you're happy, when you're writing diabetes instead of diabetes mellitus. Let's continue. They're saying that the pancreas is not producing hormone or hormone that is insulin, meaning that insulin, you know that insulin is very important for stimulation of the conversion of glucose, the excess glucose to glycogen. So automatically, if there is insufficient insulin, it means that excess glucose will not be converted to glycogen. And then now the body will have a lot of glucose. And then this glucose, the person will start to suffer from diabetes mellitus. How do you test for this diabetes in a simple way? Yeah, if the amount of if you go to you urinate and urinate, you find glucose in your urine, that could be some of the few signs that maybe diabetes is coming. I'm not saying that if you find it, you are having it, but could be the sign. Ne? Yes. So we are saying that diabetes type 1, what happens? The pancreas is not producing hormone, that is insulin, necessary for controlling the glucose level in the human. Diabetes type 2, that is diabetes mellitus type 2, in other words. Yes, insulin is available, but the body cell is unable to control the what? The glucose. So you find, we can bring diagrams like this, and then we ask you what is being illustrated in the above diagram. Yes, so that condition is diabetes. Here is a question. Here is a question, and the question is asking you to give us some of the answers which are here. Can I do bingo for you? Can I do bingo for you? Meaning that, can I give you a few seconds to do them, to do the questions, and then, okay. If you are on live, I'm giving you 1 minute to answer. 1 minute. 15 seconds. 15 seconds. So that we go through and we see that what we went through or what we have gone through is correct. What you have done is correct based on what we're going to give as the answer. Okay. 10, 8, 6, 4, 2, 0. Okay, let's go through it. Let's go through it and then we see. Okay, you're saying that you have high level of glucose in the blood. This level, yes, there is a pancreas here. Why is this organ called pancreas? Yeah, don't forget that a pancreas looks like a banana. I mean a real banana, né? The banana for gorillas. You know, gorillas and chimpanzees like bananas. Anyway, then low levels of blood glucose, yes. But here, it's still something is going to the pancreas. And then now, this release of hormone 3, release of hormone 2. See? Then now, you have increased blood glucose. And then here, decrease blood glucose. Here comes a question. And we say, organ 1. What is organ 1? We have seen it that organ 1 is pancreas. Hormone 2. Where is hormone 2? You have high level of glucose. What do we need? We need, you see, the last high. If there is an increase in the level of glucose, the last statement must be decrease. You see what I say? Yes. Increase, it means that there is more, because here they say the high level of glucose. The last statement here is low level. Why am I comparing this? Because this is dotted, it comes like that. Yes. Meaning that now the blood glucose go back to normal. So if there is high level of glucose, we need a hormone which is going to make it decrease. So which hormone is that? Is insulin. Insulin. Meaning that insulin, hormone 2 is insulin. Definitely. Hormone 3 is what? Glucagon. because if it is not insulin, it's glucagon. There is no any other hormone which is going to be talked about in regulation of sugars. Yes. So meaning that hormone 3 is, okay, is glucagon. They're saying that. Guys, there is a confusion between glycogen and glucagon. Glucagon. Glucagon is a hormone. Glucagon is a hormone. And glycogen is a substrate, is a food. Anything which has this O becomes a food, like sucrose. Sucrose. When you say sucrase, it becomes an enzyme. So it's like a hormone. It's the same thing. You talk about, maybe you talk about lactose or lactase. So when you have A, is a hormone or enzyme. A. So glucagon. I don't know whether you read it as glucagon. I don't know. Okay, doesn't matter. Glucagon, if there is A, that's a hormone. Where is O? O means food. That, guys. Food. O, food. Food. So you just know that this is not a hormone. This is a substrate. This is a food. Food. Oh, so guys, I think you no longer now messing it up. You're no longer messing it up. When are you going live for grade 11? Soon. I think on Friday and Saturday. I'll be coming for excretion. Yes. So we are saying that they are saying that disorder caused when gland 1 fails to release sufficient amount of hormone 2. So we don't have any disorder here. The only disorder we have is diabetes mellitus. Simple. The mechanism, when they talk about the mechanism, then you talk about negative feedback mechanism. When they talk about the condition, then you talk about homeostasis. So basically, homeostasis is a condition of the internal environment being kept constant. But what makes it to be constant is the negative feedback mechanism. I think I'm clear there. Is that difficult? You must remember. Ne? Yes. We will do more questions towards the exams. Ne? Towards the exams, as we are planning to be live for 24/7 from the beginning of prelim for all subjects until you write your end of the last question, the last paper in your final exam, meaning that you will be live for almost 2 or 3 months. Yes. So keep yourself on Thunder Educ so that you can obtain a distinction. All the help will be here. We are saying that the mechanism that controls the level of glucose in the blood, we say that that mechanism is called negative feedback mechanism. Negative feedback mechanism. I think I'm clear there. Is there any question before I go to regulation of carbon dioxide in the body?
Please use the link below to get the notes, questions, and other videos. Don't forget to subscribe, like.
and as the structures change, which kind of hormone are being produced. The empty Graafian follicle, and then it's being changed to corpusculum, and then now it's either will degenerate or it will continue being big, depending on whether the person is pregnant or the person is not pregnant. So here we are seeing that the female sex organ, the female sex organs are found in the lower part of the abdominal cavity, the space of the abdomen. Yes, that's the meaning. Are made up of the covering called germinoepithelium. It means that the outside lining of the ovary, we call them germinoepithelium. When you call them germinal, it means that they can have ability to give rise to other structures. Germ, it means cells, very small cells which have ability to grow with a large number of follicles. So it means that inside you have many follicles. You have many of them in germinoepithelium produced by the follicle. So now we are saying that this process, this produces, now these follicles, when they try to develop, they produce what called the ovum. This produces ova during the process called oogenesis. So production of ovum is called oogenesis, and we saw that, and the production of sperms, we call it spermatogenesis. So oo is being derived from the word ovum. When you say oo, it is derived from the word ovum. Genesis means beginning. Yeah, so it means that beginning of the ovum, or the start of the ovum. So the follicle secrete, now once this Graafian follicle develop, once it develops, as it develops, it starts to produce a structure called a hormone called estrogen. And then now, this estrogen, this follicle, once the ovum is being released, it changes to corpusculum. And then now the corpusculum will start to produce another hormone called progesterone. It means that hormone from structure A is going to be estrogen. And then hormone from structure C is going to be progesterone. It's going to be progesterone. So you have to know those two differences, that estrogen is being produced by Graafian follicle, while progesterone is being produced by corpusculum. Puberty in males. We saw, puberty in males. So if you look at this male, this means that these are the secondary sexual characteristics, the characteristics you obtain which show that now you are what? You are a mature person. Yes. What are some of those characteristics which show that now you are a mature person? So number one, puberty is the period in humans in which they are experiencing what called a physical change in their bodies in order to be capable of sexual reproduction. So if you see the following structures we're going to be talking about, it means that when you're a guy, and then you are tampering sex, you are going to impregnate someone. And then if you go to lady side, if you're a lady, and then you have these characteristics you're going to be talking about in female, then if you tamper with the sex, then you're going to become pregnant. It means that you are sexually mature. However, in the country, you are still young. It's a period which indicates sexual maturity. It means that you are sexually active. You can do sex, but don't forget to stay virgin. All right. In boys, stimulated by testosterone. So you'll find out that in male, secondary sexual characteristics, as we saw that, testosterone is the hormone which brings about this these characteristics. We said that testosterone has two functions, if we can recall. We said that the first one is it brings about maturation of sperms. Number two, it brings about secondary sexual characteristics in male. What are those characteristics? The first puberty change is an enlargement of the scrotum. It means that now the testicles are going to become big. They are starting to become big. If you compare your testicles before, when you're still young, and then now you find out that those testicles are now too big. So they start to enlarge because of the testosterone being produced, so that these testicles can start to produce sperms. As the testes and the scrotum continue to grow, what does that mean? If the testes and the scrotum they develop more, it means that now the person is going to have a big penis. Even the penis is going to enlarge. It's going to become big. I'm very sorry if you have a smaller one. So the growth of pubic hair also, these are the hair you find in on your private parts, and then you find the facial hair, hair under the armpits. Actually, you see that now a guy becomes hairy, like you have beards. Yes, it shows that now you are mature. Then another one is development of muscles and deepening of the voice. However, some people, they have to go to the gym to show these muscles to come out. However, others are born like that. Others are born like that. You can comment down below and then tell us which kind of muscles do you have. Do you have the small ones or you have the big ones? All right. And so development of muscles, it means that now this person is muscular. Is it muscular? What about the secondary sexual characteristics in the female? We see that here we have changes which occur under the influence of estrogen. We saw that estrogen perform two functions. The first one, it is very important for secondary sexual characteristics in the female. The second one, it's also thickening or development of the uterus for implantation, or it develops or builds the uterus so that implantation, or it prepares the uterus for implantation by making the endometrium, by making the uterus thick, vesicular, and granular. More thick, it must be thick. More, it becomes thick, and it becomes more blood vessels. That's why you're saying that vesicular. And then granular, it means more glands. So the growth of the female sex organ, it means that now the vagina, the things we are going to become big. It can accommodate the penis. And then you're saying that it starts the menstruation. If you see that blood coming out, the nice one, and then you just know that now you are under puberty. It means that you are develop. Then production of the ova, it means that now if you tamper with the sex, you are all going to become pregnant. And then growth of the pubic hair, that is the pubic hair that we are talking about. You find them that yes, you develop some hair, yes, here and under armpits, yes. And then growth and development of breasts. Yeah, there are some people who don't have breasts. It doesn't mean that you are not mature. Yes, you are mature. Then development of the breast, it means that now the breasts are what? Are big. Which kind of breast do you have? I don't know. All right. Then you're saying that even hips. Yeah. However, you can also find this in male. Why should male have hips? That's a lesson for another day. All right. Development of the hips so that the hips can, you can widen. Yes, you can widen. You can accommodate the developing baby. Yes. And then now, if you have seen the secondary sexual characteristics in male and you have seen the secondary sexual characteristics in female, it means that you are ready. If you go for sex, you are able to conceive. You can become fertilized. So how are these ova and the sperms being generated? Formation of gametes. Remember, when you talk about gametes, in this case, we are talking about sperms, and then you're also talking about ova. Remember, if it's one, it's called the ovum. And then if there are many, we call them ova. So this is the formation of the gametes. All right. So now you're saying gametogenesis is the formation of the gametes. Is the formation of the what? Of the gametes. In these gametes, we are saying that it includes the sperms and also includes the ovum. Then during this process of gametogenesis, the germ cell will undergo mitosis. Remember when we talk about mitosis, we are having four cells. You have one cell undergoes meiosis 1, and then also another cell undergoes meiosis 1. Yes, so you have one cell to form 2, to have one cell forms 2, and then to forms 4. It means that I have 4 cells, but all these 4 cells are haploid. They are what? They are haploid. And then this one is what? Is diploid. That's the meaning. So now, if you have meiosis, you form 4 cells. You produce haploid cells that are directly developed into gametes. Now, these cells, when it reaches meiosis, meiosis 2, these are still cells. They have not yet developed into sperm cells or ova. No, they have not yet developed. So after that, they have to undergo differentiation and then develop into cells with the specific structures. So you're saying that, hence in male, meiosis is an integral part of gametogenesis. It means that in animals, meiosis is a part of gametogenesis or formation of sperms. So this is divided into 2. Number 1, male gametes formed by spermatogenesis. It means that now the formation of spermatozoa. And then female gametes are formed by a process of oogenesis, which means that formation of the gametes in female is called the oogenesis. Formation of what? Of ova. So it means that this is formation of sperms, and then this is formation of ovum. All right, let's look at spermatogenesis in detail. Formation of sperms, the beginning of sperms. Yes, this is the beginning. This is sperms. Yes, sperms, and then this is beginning. So formation of sperms, the beginning of the sperms. Where do sperms begin from? Spermatogenesis is the formation of sperms. We have seen that. The process include, this is how we want it in exam. However, other books will talk about a lot, yeah, and detailed. But based on examination guideline in South Africa, this is how we want it to be written on what? On the paper. So under the influence of testosterone, we give you a tick for mentioning the word testosterone. Yes, the diploid cell in the seminiferous tube. You have to tell us where these diploid cells are found. They are found, the diploid cells, in the seminiferous tubes. Then we give you a tick there of the cell undergoes mitosis, undergoes meiosis. So the word meiosis is also a key point. Then you're saying that to form haploid sperm cells, to form 4 haploid sperm cells. Basically, that's what we want. And then you get 4 marks. So under the influence of testosterone, the diploid cell in the seminiferous tube, yes, undergoes meiosis to form 4 haploid sperm cells. Simple. No stress. But you'll find out that people will talk about a lot. The germ cells undergoes meiosis to produce 4 haploid cells called spermatids. And then the spermatids will mature to form the spermatozoa. To form spermatozoa. And each spermatozoa is made up of a head, middle piece, and a tail. That is the previous way how we used to explain it. But now we need it to be explained like this. The reason why I put this here is because sometimes we can bring a question and then you don't know which one is haploid, which one is diploid. Yes. So now, like this, you have the spermatogonium. These cells, they will undergo. We don't want this detailed part of it. The reason why I brought it like this, so that now you know that this is 2N, that this one, the first one is 2N, the second one is, and after meiosis 1, they become 1N. And then under meiosis 2, they remain 1N. Because 1N to versus 1N, if this is 23 chromosomes, even this one is going to be 23 chromosomes. So it behaves like as if it is mitosis. But in meiosis 1, there is decrease in the number of chromosome number. If you start with the 46, now you're going to have 23, which means that there is a decrease in the chromosome number. Structure of the sperm. How does the sperm look like? I usually tell people that a sperm is like a human. Why do I say it's like a human? Why do I say it's like a human? Remember, human has a head. Ne? A human has a chest. Ne? A human has legs. In science, we can say that is a podder. Yes, foot. So human has this. If you remove the head, you don't have a human. Sperm. Sperm has a head. Ne? Sperm has a middle piece. I'm just drawing a rough structure. Sperm has a tail. Ne? Sperm has a tail. So it means that if we call this a head, this I'll call it middle piece. Then I'll call this tail. Sometimes flagellum. So this is the middle piece. This is the head. Don't you see that it resembles a human? Yeah, it means that you come from where? Remember that a human has hair. Ne? Also, the sperm has the acrosome. Remember, a human has the brain, which controls the activities. Also, the sperm has the nucleus, which controls the activities. See, remember, a chest is for energy. Also, the sperm is for energy. Middle piece is for energy. Remember, legs are for locomotion. Also, tail of flagellum is for locomotion. So, it means that we are in a picture. We live in a picture of a sperm. That's how we live. When we see someone, it seems like it's a picture. We see a moving sperm. I'm sorry. All right, guys. So now let's look at it, the structure based on science. Sperm is made up of, number one, the head. We saw that the sperm has a head, which contains the acrosome, which looks like hair, and then that contains the enzyme to digest the what? The membrane of the what? Of the ovum during fertilization. So this, what we want is the head contains acrosome, which contains enzymes which digest the membrane of the ovum during fertilization. So when it's like an adaptation, that head has acrosome. What's the function of acrosome? Contains enzymes. What's the function of the enzymes? To digest the membrane of the ovum during fertilization. So it's like the teeth on the human, which break down whatever you want to eat. So it has the nucleus which contain the genetic material for inheritance. Whenever you talk about the nucleus, you are talking about the genetic material. Then it has a middle piece. Middle piece with mitochondria is the powerhouse. Yes, mitochondria to provide the energy. We say that it has the mitochondria, it has the middle piece. Remember, your chest is for energy. If your chest is weak, you don't have energy. If the middle piece of the sperm is weak, the sperm won't have energy. Then it has a tail which provides propulsion force for movement, propulsion force, it like for locomotion. It gives the forward force. That's the meaning. So this is how the sperm looks like. You have a sperm, you have a middle piece, and then you have a tail. Then you're saying that if we bring it in exam, ask you what is A, you have to tell us the acrosome, nucleus, middle piece, and then tail. What's the function of B? It contains genetic material for inheritance. But A, it contains enzymes which digest the membrane of the ovum during fertilization or for fertilization. So you have to know. When you bring questions, you can say, write down the adaptation of the sperm for locomotion. Now you talk about the tail, and then talk about the middle piece. And the function provides propulsion force for locomotion, provide energy still for locomotion or for movement. So they are talking about for adaptation of the sperm to carry out movement. What about adaptation of the sperm for fertilization? Then you can talk about the head, which has the acrosome, which digests the membrane of the ovum, and then you can also talk about the nucleus, which contain the genetic material for inheritance. So that is the sperm. So that is the sperm in detail. Let's look at oogenesis, and then you finalize with the structure of the ovum in detail. So oogenesis is the formation of the ovum. When we talk about formation of the ovum, we saw it. Process of oogenesis include under the influence of FSH. Yes, what happens? The diploid cell in the ovary undergoes mitosis. Diploid cell, we saw them, the germ cells, under influence of this of follicle stimulating hormone, the hormone that stimulates the follicle, the development of the follicle. The name is given, gives the function. Follicle stimulating hormone, hormone that stimulates the development of the follicle. This stimulates the diploid cell to undergo mitosis, to multiply these cells, and then only one will start to form numerous follicles. But saying that only one cell inside the follicle enlarges and then undergoes meiosis. So only there are a lot of this, but only one will enlarge to become now the primary oocyte. We just say only one cell inside the follicle enlarges and then it will undergo meiosis. Now this cell is going to start growing under meiosis 1. It means that this is meiosis 1. Yes. And then you're saying that of the 4 cells that are being produced, only one. It means that now then it undergoes meiosis 2. Yes. It is supposed to undergo meiosis 2. Meiosis 2. Yes. But now, once it undergoes meiosis 2, not all of them are going to become ovum. Only one. The remaining are going to become polar bodies. So we don't, these ones, they degenerate. They don't become big. Only one is going to become big, or it's going to develop. And then survives to form a mature haploid cell. It means that now this is 2N. And then this is still 2N because meiosis has not taken place. So since meiosis 1 has taken place, this one is going to be 1N. It's going to be 1N. Also, this one is going to be 1N. This is 1N, 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 1N. But only one will develop. So this is the one which is going to form a mature ovum. Yes. So if this is 46, this is going to be 46. It's going to be 23. It's going to be 23. It's going to be 23. It's going to be 23, 23, 23. So basically, that's the meaning. And then they're saying that one cell inside a follicle, one cell inside a follicle, yes, will undergo meiosis, and then form 4 haploid cells. The 4 haploid cells will break up, and then one cell will mature to form the ovum. So basically, this is what we are trying to explain. In the exam, this is how we bring it. Then let's look at the structure of the ovum. Structure of the ovum, you don't need to talk about the detailed part of the ovum. We want to talk about the few parts of the ovum. Parts and the function, the gel-like structure, that's the outside part of the ovum, which protect. Nah? Don't talk about the zona pellucida. No, we don't talk about those things. We just talk about gel-like layer. Yes, so it combines all those layers which are included there. So those other parts, you will see them when you go to the university. Then the haploid nucleus contain genetic material for inheritance. Haploid nucleus contain genetic material for inheritance. And then lastly, we say that whenever you talk about nucleus, you're talking about inheritance or genetic material. Then the cytoplasm, whenever I talk about the cytoplasm, we are talking about the fluid of the cell where the activities take place. Cyto comes from the word cell. And then plasm means fluid. Yeah, the fluid of the cell. Yes. Nucleoplasm. fluid, nucleus, the fluid of the nucleus. So that's the meaning. Cytoplasm, the fluid of the cell. So this is how it looks like. This is the gel-like layer. You see, this is the gel-like layer, but we have specific names for that, but we don't talk about those names. Then you have the cytoplasm. Then you have what called the nucleus. So there are 3 parts you need to know when it comes to the exams. So if they are like that, now you can name them, I think. The first one is, you can tell me, first one is, the second one is, and then the third one is. Let's see if you have got it right. The first one is the gel-like layer, the second one is the cytoplasm, and then the third one is the nucleus. Basically, that's the structure of the ovum. The menstrual cycle. The menstrual cycle. Menstrual cycle, when you talk about the menstrual cycle is divided into 2. We're divided into what called the uterine cycle and then the ovarian cycle. When it comes to the menstrual cycle, this is what you need to know. Progesterone. You need to know that these two, they come from the brain. Which part of the brain? Hypophysis, or what called a pituitary gland. So they come from the brain, and then these two, they come from the ovary. But you have to be specific. This one comes from the Graafian follicle, while this one comes from corpus luteum. Yes, but these are ovaries. So you also need to know what you need to know about that these 2 hormones, they work under ovary. They work under ovary. Therefore, we undergo what called the ovarian cycle. So whenever you ask you to explain the ovarian cycle, these are the hormones you are supposed to talk about. While these ones, they still in the ovary, but they work no, they don't work in the ovary. This one, they work in the uterus. They work in the uterus. So therefore, they are under what you call the uterine cycle. So this is the brief explanation of these hormones and how you need to know. Yes, so now you have to know the function of each hormone. Once you know the function of each hormone, then the ovarian cycle and the uterine cycle and the menstrual cycle is almost done. You need to know that in the brief, you need to know that follicle stimulating hormone is the hormone that stimulates the development of the follicle. Luteinizing hormone, this one triggers ovulation and also changes the empty Graafian follicle to corpus luteum. This one, now we are going to the uterus. This one means that it prepares the body for implantation. Yes, for implantation by making the uterus, you see, by making the uterus thick, vesicular, and granular. And then also this one is very important. In maintaining pregnancy, like building a building and then you rent it to someone, the other person will come and maintain it. So it means that estrogen builds the uterus, but the progesterone will come and maintain what estrogen has built. Yeah, you will be talking about the endometrium. It makes the endometrium wall more thicker, more vascular and granular. But we saw that the endometrium, the uterine wall, and the uterine cavity, all of them are uterus. It's a part of uterus. So now if we go back here and we see the ovarian cycle or the menstrual cycle, we shall see that the menstrual cycle is divided into 2. As we said, the uterine cycle, it means that this occurs in the uterus or this occurs in the ovary. And then you're saying that we saw this structure, the development from day 0 to day 28. And then you're saying that ovarian cycle, the development, this is menstruation, and the development of the ovarian of the uterus, then we call it the uterine cycle. So these are the 2. We have seen the hormones which affect this. You see, now the blood vessels have thickened and then the menstruation has occurred. So let's look at the events of the uterine cycle. Events of the uterine cycle. I know you guys were tired of it. Let's stop here. In other words, gland A is from the brain, and then they work in the ovarian cycle. The first one is luteinizing hormone. The second one is follicle stimulating hormone. So it means that those hormones which are going down must either be luteinizing hormone or follicle stimulating hormone. Don't forget to write the correct spelling. So you're saying gland A, structure B, follicle stimulating hormone. Basically, now we see that this is follicle stimulating hormone. If this is follicle stimulating hormone, automatically this one is going to be luteinizing hormone, because we said you only have two hormones, and follicle stimulating hormones, they have talked about it. So the next hormone must be luteinizing hormone. And then what is this? This is the mature Graafian follicle. This is the ovum, and the process which is taking place here, it is ovulation. And then once ovulation takes place, you go to what you call corpus luteum. So this is the empty Graafian follicle which has turned into corpus luteum. So basically, we have seen it. We said this is a mature Graafian follicle, and then this is the developing follicle. These are the developing follicles. All right, let's start to answer the questions, and then we see. Saying gland A, we have seen it that gland A is pituitary gland, or sometimes we can call it pituitary gland or hypophysis. Pituitary gland or hypophysis. Then structure B is here, which is the Graafian follicle. So we prefer to say mature, because even this one is Graafian follicle. Even this one is mature, is Graafian follicle. Even this one is Graafian follicle. That one is Graafian follicle. So what is the difference between this and this? So the difference is this one is mature, ready to produce the ovum. So it is mature Graafian follicle. It's mature Graafian follicle. And then structures, process C, we have seen it. Process C, we say that this is ovulation. And then you're saying that structure D, we say this is corpus luteum. Corpus. You don't forget between corpus luteum and corpus callosum. Corpus callosum is found in the brain, while corpus luteum is found in the reproductive system. So we go to the next question. They're saying that structure D, you have seen it, state the effect of estrogen levels in the blood if gland A stops secreting follicle stimulating hormone. So if they're saying the effect of estrogen in the blood if gland A stops secreting follicle stimulating hormone. So basically you have to know what is the effect. It's 1 mark, but you have to know the criteria behind. What is the effect of follicle stimulating? What's the function of follicle stimulating hormone? So the function of follicle stimulating hormone, the answer is already there, is hormone that stimulates the Graafian follicle. That's the answer already. So for the function of follicle stimulating hormone. So if the Graafian follicle does not develop, and we know that this Graafian follicle is the one which produces estrogen. So if Graafian follicle has not developed, then the level of estrogen will not be produced. Therefore, the level will not increase. It will either remain low or it will decrease, because the one which was there has been used up. So it will remain low or it will decrease. Then what state one function of LH? LH, we said it's a luteinizing hormone. It has two functions. The first one, it triggers, it causes this process to occur. It triggers ovulation or stimulates ovulation. Number two, changes the epigraphian follicle, this epigraphian follicle, into corpus luteum. So they only want one function. Then when we go to another structure, it is for a male. The other one was for a female. Now we are going to the structure for the male. You need to know how at least to label. This is vas deferens, yes. And then this is epididymis. This is the outer part of the testis, which is the scrotum. And then this is the testis. So now let's label. Let's label based on the question. Part A, we have seen it is testis. Part B, we have seen it is epididymis. And then part C, we have seen it that it is scrotum. Don't forget to write the correct spelling for epididymis. Yes, epididymis. And also the correct spelling for testis. So testis one, ne? But testes if there are many. Testis if it's what? If it's one. And then this one is scrotum. So describe the process of spermatogenesis. I explained this the other time when I was describing the paper of 2022 for an exam for June. And then remember that we are in a period of, you have to follow what you call the examination guideline of 2021. So if you follow the one for 2017, then it won't give you some of the things will be, find them changed and then not that much detailed. So here what you need, however much the book is showing you a lot of things, please follow this. If you also use our book for the distinction material, still you can find it correct. So what do you write here? The process of spermatogenesis under the influence of testosterone. So it means that that's the hormone. That's the what? The hormone responsible for development of the sperms. Under the influence of testosterone, the diploid cell, so the diploid cell, the cell which has 2N, will undergo meiosis. So also you mentioning the word meiosis, we shall give you a tick. Found in the seminiferous tube. We need to know where is it found. Found in the seminiferous tube to form a haploid sperm cell, or to form haploid. So basically you have one cell which is 2N will undergo meiosis. Here I remember that you form two kinds of meiosis. Meiosis 1 and meiosis 2 will undergo this. Under the influence, this cell will undergo meiosis. But what causes it to undergo meiosis? It is the testosterone. That's why, where is this testosterone and this cell found? They're found in the seminiferous tubes. How do we call these cells which select themselves out to undergo meiosis? We call them germ cells. What do they form? They form the sperms. So basically, that's it. The germ cell will undergo meiosis under the influence of testosterone in the seminiferous tube to form a haploid sperm cell. Or you can say under the influence of testosterone, the diploid cell, that is the germinal epithelium, will undergo meiosis to form the sperm cells in the seminiferous tubes. So we need to know the hormone. We need to know what will undergo meiosis, that is the diploid cells. The beginning is 2N, and then where is it found? It's found in the seminiferous tube, and which process will undergo, which is the process is meiosis, and what will they form, which is what? Which is the sperms. That's what we want in this regard. If they ask you to explain spermatogenesis, the same story when you go to oogenesis. Then we go to another question. Explain why a doctor would advise the man to wear underwear that is not tight. So remember that whenever you are tight, the pressure increases. And the moment the pressure increases, in most cases, the pressure is associated with the high temperatures. And then if the high temperatures are there, then definitely the temperatures are going to exceed the normal temperatures at which sperms are being produced from. Therefore, it means that it's going to lower the sperm count. That is the basics behind that. But now, how do you answer that in the exam? Maybe you can say that the tight underwear will put testes close to the body. When you put it in tight underwear, it will pull the testes close to the body. Yes. And then this will cause testes to have high temperatures or too much temperatures. Yes. And then this will affect the sperm maturation negatively. Don't say affect the sperm maturation. How? Negatively. It means that it will lower. Yes, it will lower how many and the rate of these sperms maturing. So if there are less sperms which are being produced, it means that the number of sperms at the end will be low. That's why we are saying that it's going to reduce the sperm count. Or you can explain it in any other way, as long as you talk about that the testes are going to be a little bit close to the body. And then once they are close to the body, they're going to have higher temperature, more than the temperature needed for the sperm production. And then this will influence the sperm maturation and production, and hence the low sperm count will come in. So please don't forget to explain that. Then they're saying that during vasectomy is to cut these testes. When you cut this vas deferens is what you call vasectomy. So they're saying that during vasectomy, explain one reason why a man who, number one, does not want to have children will choose to have vasectomy. This vasectomy does not mean that if your vas deferens are cut, you cannot undergo sexual intercourse. You cannot have sex. It does not mean that, because the penis to erect is not connected to the testes. The penis to erect is connected to the blood being supplied to the penis. This person, the blood will be still being supplied the tissues of the penis. Therefore, this person can erect and can have sex. If you cut the vas deferens, the only thing vas deferens is going to do is that the semens which are being produced, remember we have the gland of prostate gland, the cowpass gland, and the seminal-vesicle, they will produce a fluid, and that fluid will not have sperms. So there will be no sperms, but that fluid will be there. The fluids will be there, but it will not have what? Sperms. And if the sperms are not produced, so what will happen? It means that the semen which are produced cannot fertilize the ovum. Hence, there is no child. But the person can still have the what? The sexual intercourse. So what is the answer in the exam? You can say that there will be no sperm in the semens, therefore no fertilization will take place. So no sperm, no fertilization. No sperms, no fertilization. So we will have no fertilization. Yes. So if you don't have sperm, no fertilization. Part B, had a vasectomy is still capable of ejaculation. Why is it that this person is able to produce other fluid from the body when you are ejaculating? Is because I've told you that you still have the other glands. Look, so vasectomy is here, and then the glands are here, and then the penis is here. So you have the vas deferens, you have the Cowper's gland, and then you have the prostate gland. So other fluids will come from those glands, and they will make the ejaculation possible. Hence, you can still enjoy sex. All right. Question 12. They said another question is about still reproduction. The first one was about female. The next one is about male. Now this one is both male and female. All right. They're saying diagram below represents structures of an ovum and the sperm. So you have to know how to label. At least to try. What is this? This is the jelly-like layer. What is this? This is the cytoplasm. What is this? This is the acrosome. What is this? This is the nucleus. What's this? It's a middle piece or mitochondria. And then what is this? This can be a tail or flagellum. So identify part A. They have said it. Jelly-like layer. Part B, cytoplasm. Part C, acrosome. You have to know the function of each. Name the process involving meiosis that lead to formation of the ovum. You see, now they want formation of or production of the ovum. So because it is ovum, so it's gonna be oogenesis. And then I'm saying write down only the letter of the part of the sperm that enters the ovum. The letter of the part which enters the ovum. So basically, if you look at this sperm during fertilization, this part will remain out. And then this part will be used to digest the membrane of the ovum. It is only this nucleus which will enter the ovum, and then the haploid nucleus of the sperm will fuse with the haploid nucleus of the ovum, and then a diploid zygote or diploid zygote is formed. So basically it's gonna be the nucleus, and which part is that? Is part D. Part D, yeah, is the one which is gonna be used. Then they're saying that write down only letters of the parts that enable the sperm to move towards the ovum. So if we look at what provides energy, we shall see that the middle piece and the flagellum or the tail are the one which are used. So the middle piece will provide mitochondria, and mitochondria is responsible for energy. And then this one, it will provide propulsion force, the forward force, so that now the sperm can move forward. Yes. Then they are saying that now we are going to process of fertilization. The schematic diagram below shows a human ovum that is about to be fertilized. The diagram is not drawn to scale. All right, they're saying what is part A? We saw that part A is the jelly-like layer. What is part B? Remember, this is part B comes up to here. Yeah, up to there. That darker layer. Remember, this is a cell. And all cells, they have a membrane which protects them, allow gases, substances to go through, which we call cell, the membrane of the cell. The membrane of the cell, which we call cell membrane. So The nucleus is there. Remember, this is an animal cell. Therefore, B becomes the cell membrane. What is C? C, you have the fluid there, which we call cytoplasm, the fluid of the cell. And then what is D? D, you see that is the nucleus of the sperm. Nucleus. And then what is E? E is the acrosome. And then what is F? F is the nucleus of the ovum. And then what is G? G is the middle piece. Middle piece. So basically we have labeled it. So A, they want A, we have seen it. A is the gel-like layer. B is the cell membrane, the membrane of the cell, or plasma membrane. Then C is the cytoplasm. And then F, where is F? F is the nucleus of the ovum. So now give the letter and the name of the part that contain mitochondrion. We have seen it that mitochondrion are being stored in here. If one is mitochondrion, if there are many, so the answer is G, and the part is middle piece. Contain enzyme required for penetration of the sperm into the ovum. So it is this. It has the enzyme. We don't need to know the name of the enzyme. Then the part, we are saying that it is E, and the part is acrosome. You don't need to know the enzymes because this is not our syllabus. And then they're saying D, C will enter the ovum during fertilization, and that is the nucleus of the sperm, which is part D, nucleus of the sperm. And then here you have the structure of the sperm. You're saying that A, B, C. Now we have tried to answer questions concerning about the sperm. So there is no way they can bring a question you have never seen about the sperm, about these structures of the sperm and the ovum. So we're gonna go to even the graph. We also explain. We're gonna explain all topics in such a way that there is no way you can fail this. There is no way. And if you do this, you go back and listen to this before the exam, before you write the exam, look, and at least you are reading like this, then automatically just guarantee yourself a distinction. All right. Then they're saying that what is part A? We have said acrosome, part B, nucleus, part C, middle piece, part D, the flagellum or the tail. They are saying that identify B. We have seen it. We have seen it. It is nucleus, part D. We have seen it is the tail. Then they are saying that explain one way in which the sperm cell is adapted to ensure effective movement. Whenever they talk about movement, you talk about the parts which help the sperm to move. And we only have two parts which help the sperm. Adaptation is the part and the function. The first one is the tail. Yes, the tail. This provide the forward force, or provide the propulsion force. So it allows movement to take place. So basically this is for force. And then number two is the middle, this part, middle piece, middle piece. This one provides it has mitochondria which are responsible for production of energy. Yes, production of what? Energy. Explain the consequence for reproduction if the sperm did not have part A. What is part A? It is the acrosome. So what is the function? You first ask yourself, what is the function of the acrosome? The function of acrosome is to release enzymes which are used to digest the membrane of the ovum so that the sperm can penetrate during fertilization. So the major function is to contain enzymes which digest the membrane of the ovum so that the fertilization can take place. So if part A is not there, it means that that will not take place. It will not happen. So basically, no acrosome will be present. So the word no acrosome, yes. And then therefore there will be no enzymes present, and the sperm will not be able to penetrate the ovum, and therefore no fertilization. If there is no acrosome, there is no enzymes, and the sperms will not be able to penetrate the ovum, and no fertilization will take place. At least you will have something like 4 marks out of 3, and then we'll be able to obtain those marks. Guys, please watch these videos. Forward them to others so that they can also benefit. If you have a question, we are here to try to answer those questions, because some of these questions we do as a result of other students trying to ask for help. Now you have another question.
Please use the link below.
To the sperm, and the mother gives out to the ovum. These two, they contain the genetic material which provide genetic material for the incoming generation. So when the fertilization takes place, then conception is being done. So that is the time when the male gamete fuses with female gamete. Then we say that you are pregnant or you have conceived. So pregnancy begins immediately, or once the fertilized egg is implanted in the what? In the uterus. The moment implantation occurs, then it means that we say that the person is pregnant. But implantation cannot take place unless fertilization has occurred. So this is what you call the outline of human life cycle, from the time of sperm and ova being produced to the time of maturity, so that these individuals can produce the ovum and the sperm again, so that another individual is being formed. So in the human body, constantly develop and the changes occur throughout the life. And the food provides the fuel, or which we call the nutrients. So this fuel is what you regard as nutrients. So whatever you eat, when you're hungry, you feel weak. With the moment you finish eating, you feel energetic. It means that the food you have eaten has provided fuel to you, so that now you can do other body activities. In the life cycle, you'll find out that you start with the ovum and the sperm. When these two fertilize, when they fuse, the haploid nucleus of this sperm, when the haploid nucleus of this sperm, yes, fuses with the haploid, haploid nucleus of the ovum, then you form a zygote. So fertilization occurs when these two fuse, you form a zygote. Remember, a zygote is diploid, and then mitosis occurs. It means that you multiply these cells. You multiply billions of cells, meaning that all these cells are identical as long as they have not undergone differentiation. So now the moment the adults form, now they produce sperms from the testes, ova from the ovary, and then meiosis occurs again, and then sperms again being produced. So the cycle continues like that. So we saw that meiosis is very important in bringing half the chromosome number, because we say that this is diploid. So, and then the sperm is haploid. You see here meiosis. Meiosis, once it occurs, it breaks down, or it divides these cells into haploid. Also, it is on the haploid, so that fertilization can take place. And then the haploid and the haploid, they fuse, which we call the doubling effect, to form a diploid. And then the cycle continues like that. Do you know about the male reproductive system? Yeah, many students know about these structures, but sometimes when you bring it in exam, you find out that students can't even label their own structures. That is an embarrassment. So let's look at the male reproductive structure. The male reproductive structure. There are some specific parts which you need to know and understand. So the male reproductive structure is made up of what called scrotum. Scrotum is the outside part. This is the scrotum, the outside part, which keeps the testes. We call it scrotum. Then the second one is what we call the testis. So the scrotum covers what we call the testis. We're going to see the functions in detail. Then we have what we call the epididymis. Epididymis is this coiled part you see. This coiled part tries to store the sperm so that they can mature. Then we have what you call, now when you go like this, we have what you call the seminal vesicle. Seminal vesicle is very important in providing a specific fluid. We're going to see that specific fluid and its function. Then we have what you call the vas deferens. Vas deferens is the tube which gets the sperms from the epididymis and then takes it to urethra. So this is what we call the vas deferens. And then we have what you call another. This is a first gland. We call this one the first gland. This is the first gland. Then we have the second gland, which we call the prostate gland. It's also another gland. We're going to also see the function. Then you have the third gland when you move out. So if you follow this, you'll find out that the first gland is seminal vesicle. The second gland is prostate gland, and then the third gland. So I will call this one the third gland, which we call the Cowper's gland. So now you have what you call bladder. Bladder is not a part of reproductive system, but we can call it a part of urogenital system, urinary system, and then the genital, which is the reproductive system. Yeah. But we bring it so that we can show you exactly the position of the bladder versus the position of the prostate gland. Because sometimes students confuse the prostate gland with the bladder. Then we have what we call the ejaculatory duct. These are ducts. This is where these other glands releases their fluid in. Releases their fluid. So we call them ejaculatory ducts. Then we have what we call a penis. Penis, obvious. Everyone knows about the penis. I don't even need to explain. However, we shall see the actual function of the penis, because sometimes some people, they misuse the penis because they give it another function which is not supposed to be. All right, let's look at then the urethra. Inside the penis, there is a tube which brings out the sperms and the urine and all the stuff which is coming from inside. We call it urethra. And then lastly, we have what the foreskin. Foreskin basically is meant to protect the top part of the penis, but it brings so many diseases. So we prefer to cut it, and then we say that you have done what we call circumcision. But we shall see the function of each part. So we call this one a clear view. So when you look at the clear view, this is what you see, exactly what you see. The penis, urethra, you go to the prostate gland, you go to the seminal vesicle, Cowper's gland, it's supposed to be here, and then the tube connects to the epididymis. This tube is called the vas deferens, and then it comes to the epididymis, and then you have the testis there. Then the functions, let's look at the detailed part of the structure of the male reproductive system, which called the functions of each part. The question can come in, why do testes hang outside the female body, the human body? Okay, we shall see that answer. So the first question, the first part is, we say that the testis. What is the function of the testis? The testis has two functions. The first function is to produce sperms. The second function is to produce testosterone. Two functions of the testis. The first one is production of sperms. The second function is production of testosterone. Testosterone is for secondary sexual characteristics. Sperms is those ones which carries genetic material to the offspring. Scrotum, which is the outside part of the male. Scrotum is a skin sac that protects the testis, and then you hold the testis, yes, outside the body at a temperature that is 2 degrees below the body temperature. So 2 degrees below the body temperature. Why is testis supposed to be kept to a temperature less than the body by 2 degrees? The body temperature is, we know that the body temperature is 37. Yes, so if it means that it's 37 or 37.5, then it means that the testis temperature should be around 35. Why? Because this is the temperature which is good, favorable, optimum temperature for the sperm to be produced or for sperm production. So this is the best temperature for production of the sperms. Then another part is what called the epididymis. Epididymis is this coiled part. Is this coiled part of the testis? This coiled part of the testis. This is very important in storing the sperms so that they can mature. When the sperm are being produced, they don't mature immediately. No. Actually, sperms spend almost 3 months to get mature. But every day, a human body produces over 100 million sperms. Over 100 million sperms every day. But it means that the process, it started way back. So that by the time every day, over 100 million sperms are being they are being released into the epididymis. Then the epididymis store them, and then they get mature or they mature. After that, they can be released to carry out the function of fertilization. Yes. So epididymis, the function is to store the sperms so that they can mature and then ready to carry out fertilization. Then we have what called the vas deferens, this tube. Sometimes they call it sperm duct. Don't call it sperm duct because you'll cross it. Its name is vas deferens. When you call me a teacher, it doesn't mean that that's my name. That's not my name. So you call me a teacher because of the job I'm doing. So when you call it sperm duct, that's not its name. It's because of the job it's doing. Sperm duct. Sperm, you know the sperms, and then duct means a tube where sperms passes. So they call it sperm duct, but that's not its real name. Its real name is vas deferens. So in exam, we want you to write vas deferens, not sperm duct. So it transports sperms from the epididymis, from this epididymis where they're being stored, to the urethra, to the penis, where the inside part of the penis, so that now the sperms can move to the vagina. Then we have what called the Cowper's gland. Cowper's gland. This is Cowper's gland. This produces mucus. We say that we have 3 glands. Gland 1. We say we have the gland 1, gland 2, and gland 3. These 3 glands, we call them accessory glands. So the first gland from outside, we call the Cowper's gland. This Cowper's gland produces a mucus. When you talk about a mucus, you are thinking of rubrication. It rubricates, and then it helps in the movement of these sperms. It rubricates so that when the sperms are coming, they are moving at a very high speed. If you can check how much speed the sperm they are moving when they are moving out of the penis, the speed is very high. That's why the gentleman immediately after ejaculation loses energy because of the speed and the energy these sperms are using when they are coming out of the penis. And then also the contractions which are happening, because this speed is being brought about by the contractions of the muscles of the body, and then because of the too much energy which is being lost, then it ends up the individual loses the energy at the time of ejaculation. All right. Then we have what called the secondary accessory gland, which we call the seminal vesicle. So seminal vesicle is very important in production of nutrients. We say that the sperms are supposed to have energy, but how will they have energy if they don't have nutrients? So it's the function of the seminal vesicle to produce these nutrients. These nutrients, they come as fructose. Fructose are sugars. So these sugars, they contain a lot of energy, and then this energy can be used by the sperms to swim towards the ovum. So we're going to see it, the structure of the sperm. How does it use the energy being produced? And then we have the last accessory gland is called the prostate gland. This produces the alkaline fluid that neutralizes. We know that the urethra of the penis is acidic because of the urine which passes through the penis. And then we know that the vagina is also acidic. Yeah, because sometimes it can be too acidic, and then in such a way that it kills most of the sperms, and then it can prevent fertilization to take place. So if that situation happens, it means that the lady is supposed to go to the gynecologist, and then they give her some recommended medications to reduce the acidity of the vagina, so that not all the sperms which come into the vagina are being killed. So the prostate gland, the function is to provide this alkaline fluid. This alkaline fluid tries to neutralize the acidic nature of the vagina and the urethra or the penis when the sperm are being moved. Why? Because sperms don't require acidic medium. If the acidic medium is present or is too much, then it's going to kill these sperms. So the acid is produced in the vagina, which would kill the sperm cells. So if the acid is too much, then the sperm cells are going to be killed. That's why we produce what we call the alkaline fluid to neutralize this acidic nature of the vagina and the urethra. Then the urethra, what's the function of the urethra? When you talk about the urethra, as if you're talking about the penis. Why? Because the urethra is the inside part of the penis, is the tube which passes through the what? The penis, where urine and the semen are being produced or passes through when they're coming out of the what? Out of the body. So urethra transports semens. Now I've talked about semens. What are semens? Some people confuse what calls semens and sperms. So these are two different things. When we talk about sperms, sperms are what called the spermatozoa. But if you talk about semens, we have seen that these three accessory glands produce some fluids. These fluids, the fluid from the seminal vesicle, the prostate gland, and the Cowper's gland, they join. When they join, we form what we call semens. So a semens is a combination of these three fluid from the accessory glands plus sperms. So when you talk about sperms, it means that there is no fluids from these accessory glands. So when you talk about semens, it means that there are three fluids from accessory glands plus the sperms. So now when you talk about urethra transporting the semens and the urine, it means that it's transporting these fluids which are coming from the accessory glands and the sperms in general. All right, let's go. Functions of the testes. What are some of the functions of testes? Testes produce a hormone called testosterone. This hormone has specific functions in the male reproductive system or in the male body. So what are some of the functions of testosterone? This is very important in the development of this male secondary sexual characteristics. These are the characteristics which shows you that you are now a man. That's why you're saying male reproductive, male secondary sexual characteristics. The characteristics which you are not born with, they come after you have grown up, and they come as a result of this hormone called testosterone, such as beard, pubic hair, deep voice, and then muscular body. So it means that if you are a man and you don't have that, does it mean that you are not a man, or does it mean that you don't have testosterone? You are a man, but you might be having less testosterone. Yes. And then you have number 2 is to stimulate the maturation of sperms. Sperms get mature as a result of testosterone. So if testosterone is not being produced, it means that the sperms are not going to mature. Then lastly is, we can say that seminal vesicle, prostate gland, this is a knot. You need to put this as a knot, This is a knot. Seminal vesicle, I explained it, is seminal vesicle, and then prostate gland, and then Cowper's gland. All these three, we call them accessory gland. We can bring this in the terminologies. And I ask you, the accessory glands, no, seminal vesicle, prostate gland, and Cowper's gland are referred to as, then they are referred to as accessory glands. Or the glands in the male reproductive system, which produce the fluids, are referred to as accessory glands. So where are the sperms being produced from? Where are the sperms being produced from, which we call the seminiferous tubes? So let's look at the seminiferous tubes, the simple structure of seminiferous tubes, and the things which you need to know about the seminiferous tubes. The first thing you need to know about the seminiferous tube is the cells of Leydig. Cells of Leydig. What is the function of the cells of Leydig? This is very important for production of testosterone. We have seen the function of testosterone. That testosterone is very important in secondary sexual characteristics and also very important in the maturation of the sperms. So testosterone, it originates or it comes from being produced from cells called Leydig, the cells of Leydig. The second cells are called satellite cells. What is the function of satellite cells? This is very important in the production of nutrients so that these sperms can develop. So these nutrients are found in the seminiferous tubes. They're very important in making these cells. They're very important in making the spermatozoa. They're not yet spermatozoa. They only become spermatozoa when they have matured. So satellite cells produce nutrients so that these sperms can develop into mature spermatozoa. So you need to know those two cells, the cells of Leydig. We like to ask you that, and then satellite cells. So if you look at the simple structure of seminiferous tube, yes, you find that the satellite cells are found here, and then these satellite cells are the one which produce these nutrients, and then these sperms, they start to develop. So cells of Leydig are here where they produce the testosterone, and then this testosterone which is being produced here, they are very important in making these cells to mature. Yes, so basically that is a simple structure of the seminiferous tube. So we say that if we do a small recap, Leydig cells produce testosterone. Testosterone is very important in the maturation of the sperms, and then the satellite cells produce nutrients which are going to help these sperms to mature because they require nutrients for them to mature. And then lastly, let's just look at a simple structure of a female reproductive system. Female reproductive system. Female reproductive system is a little bit simple compared to the male reproductive system. And sometimes I wonder, you find out that males know the female reproductive system more than the female, and the female know the male reproductive system more than the males. And I ask myself, why? I think you know the answer better than me. All right. All right, the female reproductive system has what's called a fallopian tube. Fallopian tube. Not oviduct. The same thing, sperm duct. No. We call it fallopian tube. That's its name. Then it has what's called the uterus, where the baby develops from. Then it has what's called the endometrium, where attachment takes place. We're going to see this in detail. Cervix closes the vagina or separates the vagina from the uterus, and then gives some protection of the cervix, gives some protection of the uterus. Then you have the vagina. Simple, nice, and no need to explain. This is called a birth canal. That's where most of people or most of us passed when we are coming to this troubling world. All right. Then we have what's called a vulva. Vulva is the outside part of the vagina. So these are the parts which you need to know regarding to the female reproductive system. We will explain this more detailed and then see exactly using animals reproductive under our new YouTube channel called The Bride Doctor. So, all right, let's look at the functions of the parts. The vagina receives the penis. So the function of the vagina is to receive the penis. So it means that it's the only vagina which is supposed to receive the penis based on the science and based on what science tells us to do. So it receives penis and semen. Semen is we say that is the combination of the fluid from the 3 accessory glands plus the sperms. So we call them semens during sexual intercourse. So we have seen it here that the vagina receives the what? The penis and also the semens. So semens are supposed to go into the vagina and also based on this, and also vagina also receive the what? The penis. However, some people, they put these things in different areas. I won't talk about that. Hey, number 2 is ovary. Ovary is very important in production of egg cells. It does not stop there. It also produces what you call progesterone. Doesn't stop there. It also produces what you call estrogen. So ovary has 3 functions: production of egg cells, which called ovum or ova, and then production of progesterone, which maintains the pregnancy period, and then production of estrogen, which prepares the body for implantation. It prepares the body, the uterus, so that implantation can take place. We shall see that in the next class. Then number 2, we have what called the fallopian tube. Fallopian tube connects the ovary to the uterus. It connects the ovary. If you look at this, it connects the ovary. Yes, this is the fallopian tube. So it connects this uterus and then the ovary. So that's the function of the fallopian tube. However, the major function of the fallopian tube is the site. Is a site for fertilization to take place. It's a site where fertilization takes place. We love this in exam. When we ask you what is the function of the fallopian tube, it's a site for fertilization. Then number 3 is the uterus. This carries the embryo during pregnancy. It carries the embryo. That's where is the space. If we talk about the space, we call it uterine cavity. Yes, it has, the uterus is divided into 3: the uterine wall, the uterus, the uterine cavity, and then endometrium. Endometrium is also a part of the uterus. Yes, but now if we label them differently, then you have to understand, or you have to tell us based on where the arrow is coming from. Basically, this is the uterine cavity, and then this is the uterine wall, and then this is the endometrium. Uterine, this part is the uterine cavity, and then this is the uterine wall, and then this is the endometrium. So that's it. And then number 2 is, we say that it's very important for carrying the embryo and then the fetus. Why do we call it embryo, and when do we call it fetus? When do you call it embryo, and when do you call it fetus? We shall see it when we go to pregnancy. All right, endometrium. Endometrium is very important. This is the endometrium. So basically it's supposed to go and then attaches itself there. That's the endometrium. Endometrium is the inner lining of the uterus. That's why I said that the uterus is divided into 3: the uterine cavity, endometrium, and then the uterine wall. Yes, the inner lining of the endometrium, uterus, and then it's a place where embryo implants itself. Actually, that's where the implantation takes place. Finger-like projection takes place from there. It is where the placenta is supposed to connect. Yes. So it's very important for implantation to take place. Cervix. Cervix is the lower narrow part of the uterus, and it stretches during pregnancy to allow the baby go through during birth. And also it gives out protection because it closes. It only opens, it gets what called a plug. It only opens during around day 14 when the fertilization is supposed to take place. Yeah, that's when it tries to open so that the sperms can enter the fallopian tube, and then fertilization can take place, so that the sperm can enter the uterus and then the fallopian tube, and then fertilization can take place. Vagina, we say that vagina, it has 2 names. It's the vagina and then the birth canal. The vagina receives the penis, we said it, and then we call it birth canal because of the function of the second function of the passage where the baby passes when you are giving birth. However, some people, they get what you call C-section, and then the baby doesn't pass through the vagina, and then it passes canal, another channel which they have created. So those are the functions. And then the vulva is the external opening which allow penetration to occur. That's where the penis passes before it reaches the vagina. So that's what we call the vulva. So basically, those are some of the functions of the male reproductive system and the female reproductive system. So I think I'm going to stop here. When we come back again, we're going to talk about the structure of the the ovary, the structure of the sperm, and how do we ask questions concerning about that, the structure of the ovum, and how do we ask questions about that, and then also puberty, so that I finish that, and then I'll go to spermatogenesis and oogenesis. Then we shall end up with the menstrual cycle. Thank you very much for watching. The diagram below shows the part of the brain. Obvious, you can see it. That's the part of the brain. Okay. What are they asking? They are saying that identify part A. What is this part A? You can identify all of the parts. For example, part A is cerebellum. Cerebellum. Part B is medulla oblongata. Part C is pituitary gland or hypophysis. Part A is hypothalamus. Sorry, this is hypothalamus. And part D is cerebrum. Now, state 2 function of part D, which is cerebrum. It has 3 functions. The first function is the center for higher thought processes, which is memory, judgment, and reasoning. Number 2, it coordinates all voluntary actions. For example, raising the hand, sitting, standing, things you do on your own will. And then lastly, it receives and interprets all the sensations. For example, seeing, hearing, tasting, all that. Those are sensations. So those are the functions of cerebrum. Name the hormone that's stimulated by gland C that has an effect on, number 1, long bones. That is growth hormone. It is growth hormone which has the effect on long bones. It brings about the growth of these bones. And then now they're saying that mammary glands, that is prolactin. Prolactin, if you look at a hormone called prolactin, lactin, you know the word lactogen from ShopRite. That's milk. So lactin is milk. So lactose is milk sugar. So anything related to lact, it means that is milk. You must think about milk. So the answer is prolactin. And then they're saying that state 1 way in which the brain is protected. It's protected in 3 ways. By the 3 layers, which we call the meninges, the cerebrospinal fluid, and then also by the cranium, the cranium. So it is this cranium, not the whole skull, the cranium, because the skull, when you talk about the skull, it includes also the jaws. So you better use the word cranium. Remember, when you're talking about the brain size, how is the brain size? And the cranium, we said that don't talk about skull, talk about cranium. Then they're saying that describe the role of hypothalamus in regulating. How is hypothalamus able to regulate thermoregulation? Remember that how does it control thermoregulation? What is thermoregulation before you know the function of hypothalamus in this regard? Thermoregulation is when the temperature is being kept constant under a narrow range. So how is the hypothalamus able to do this? So when you talk about hypothalamus must work together with the skin. How? Now, this hypothalamus, it will receive and interpret. It will receive, and once it receives, it will interpret the impulse being received. And then, where is this impulse coming from? From the receptor. Where the receptor, it could be the skin or in the blood vessels, depending on where the temperature is, but mostly on the skin. So it receives and then interprets that impulse obtained from the receptor, which is the skin, and then sends the impulse to the blood vessel. So when the blood vessels, when it sends the impulse to the blood vessel, it will depend on whether the blood vessel will constrict or it will dilate. because they said thermoregulation. They didn't say that in cold conditions or in hot conditions. So when it sends the impulse to the blood vessels will either constrict or will dilate. And then it will not stop there. Yes. It will also influence how much blood is flowing under the skin. So if it is dilated, it means more blood. If it has constricted, it means that less blood will flow under the skin. It does not stop there. Remember, thermoregulation involves the skin and the sweat gland. So now it will also influence the sweat gland, either by the sweat gland producing more sweat or by the sweat gland producing less sweat. So it will influence the blood vessel, the blood which flows on the skin or in the skin or under the skin. And then also it will also influence how the sweat gland will work. So basically you can say that it receives and interprets impulse from the receptor, that is the skin, and also now it sends the impulse to the blood vessels, either to of the skin, either to constrict or to dilate. You understand? And also influences the sweat gland, either to be more active or it becomes less active. Basically, that's what you need to know, because they didn't ask you whether it is cold conditions or in under hot conditions. Part B is involved in homeostatic control of the carbon dioxide concentration. Yes. State the location of the receptor that is stimulated by the increase in the carbon dioxide concentration in the blood. Where is part B? Let's first see what is part B. Part B is the medulla oblongata. So what is the receptor? Where is the receptor? The receptor are found in the blood vessels, which we call carotid artery. Carotid artery. These are blood vessels. Carotid. Yes, carotid arteries. These are arteries. Where they found? You are found at the upper part of the human. Basically, the neck, the chest, the neck. Yeah, moving from the end of the neck to the chest to the part of the head. That's the lower part of the head. Yeah, we call them carotid artery. So that's where the receptors are found. And then B, they're saying that name the two effectors that part B sends impulse to. Where is part B now? Part B or receptors. If you look at it, there are two things which it sends impulse. It sends impulse to the heart so that the heart can either increase or decrease. Basically, the heartbeat increase so that it sends blood to the muscles and then picks up carbon dioxide. Number two, the diaphragm and the intercostal muscles. Diaphragm is also another tick, and intercostal muscles, so that the breathing rate can increase and the depth can also increase so that carbon dioxide is pumped out of the body. Carbon dioxide, we don't need it in the body. That's why it's supposed to be pumped out. We don't have the other way of the negative feedback mechanism for it. Question 3.4. This is the structure of the ear. You need to know exactly what is this. They are saying that the diagram below represents the part of the human ear with the middle ear infection. What is the A? You need to know pinna, auditory canal, tympanic membrane, the oscars, because there are three, semicircular canal, oval window, and then a eustachian tube, which is filled up with the fluid. So that's why it is middle ear infection. Identify part B. We've seen it. We have seen it, which is auditory canal, and then part D, which is oscars, which are the three bones found in the ear. State the one function of A. This one is collect and direct sound waves to the auditory canal. It collects sound waves from different directions, and then does not only collect, it directs all of it which has come there. It's being directed to what? To the auditory canal. That's the meaning. And then saying that another question is
Explain how the middle ear infection could affect hearing. How is this middle ear infection affect hearing? Which is it means that there will be a problem with the part D, these oscars to move freely. So basically, you can say that part D, or what you call the oscars. The oscars will not vibrate freely because of the fluid which is inside there. Will not vibrate freely, and then this will cause the over window not to receive enough vibration. So the over window, because this one is not vibrating freely, the over window will not receive enough vibration, and then there will be no pressure which is being built inside the cochlea. So if this one is not moving, then there won't be enough pressure which is built inside the cochlea. And then what happens? It means that there won't be enough impulse which is going to be sent to cerebrum for interpretation. Hence, this person will become deaf or hearing loss. Remember, deafness is which can be partial deaf or complete deaf. So basically, we are just explaining the transmission of the sound in the negative way. There won't be enough movement of the oscars. There won't be enough pressure built in the movement of the of a window, and then there won't be enough pressure built into the cochlea, and less impulse or no impulse will be sent to the cerebrum for interpretation, and then this will cause the person to be deaf. And then another question, they're saying that name the small device which is being used to treat the middle ear infection. Remember, this device to be used is supposed to cut here, and then we suck all the fluid which is inside here. We suck it out, and after that we put that small device so that it is able to drain and make the middle ear to be dry. So we call them grommets. So we call them what? Grommets. This grommets is not supposed to have the E. Yeah, we call them what? Grommets. Remember, middle ear infection is being caused by a bacteria, so you can also apply antibiotics. But they didn't ask you that question. They only asked for the device. Then they're saying that, oh, I've skipped one question. Describe the role of eustachian tube. The role of eustachian tube, remember, eustachian tube connects to the mouth. So what is the function of the eustachian tube? The function of the eustachian tube, it is to equalize the pressure on the either side of the tympanic membrane. So this is the eustachian tube, which connects to the mouth, is to equalize the pressure on the either side of the tympanic membrane. Then lastly, but not the least, they're asking you to write the letter of the part which where the small device named above, which is the grommet. We say that these are grommets, which is where is it supposed to be? Where is it supposed to be inserted? It's supposed to be inserted in the tympanic membrane. And then if you look at this, it is part C. And then lastly, describe how part E. What is part E? The semicircular canals. Whenever you talk about the semicircular canal, we are talking about balancing. So describe how part E is involved in maintaining balance when there is a change in the speed and direction. Whenever you talk about speed and direction, now you're talking about the cristae. Whenever you talk about this, cristae, or which is called the cristae. Remember, the other one is the maculae, which is the position of the head. So meaning that change in the speed and the direction of the head will stimulate the cristae found in the, where is cristae found? So cristae is found in the ampulla of the semicircular canal. Yes. And then the impulse which is being received is converted. The stimulus which is being received is converted into enough impulse which moves along the
auditory nerve to cerebellum for interpretation to restore balance. Or you can say that crystalli are stimulated and then converts the stimuli into enough impulse, which is sent to cerebellum, where an interpretation, and then this sends the impulse to the muscles, and then balance is being restored. Lastly, let me show you from our booklet, and then you see exactly what I'm trying to say. You see, here is the Saidi's model of balance, whereby it's saying that change in the speed and direction. You see, it is crystal found in the ampulla of the semicircular canal. The stimuli is converted into enough impulse, which is sent along the auditory nerve to cerebellum for interpretation to restore balance, meaning that sends back the muscles, and then balance is being obtained. So basically, that's what you're supposed to explain. So it means that you're looking for this route. So the paper really was also very easy, paper 1. I'll come back again to discuss other papers. Please don't forget to subscribe, stay tuned, and whenever we post, please try to watch so that you at least you upgrade your marks and then you get a distinction. Thank you very much. See you again. Okay, they are saying that the diagram below shows the eye under different conditions. Okay. All right. What makes them to be different? What makes them to be different? Okay. What makes them to be different is the lens. You see the lens here? Yes. And also the lens. However, this, and this, these are two different conditions. No, yeah, it's the same condition. This and this are also the same conditions. However, this is too thick. It's like showing a near object, meaning that it's more convex. Less convex is showing for the far object. Yes, these two. While this space shows that more light is entering. So it could be for dim light, while this one shows that less light is entering, which could show that is for dim light. It means that this person is dim light near object. Dim light, no. Bright light near object, bright light near object, and then dim light, far object, dim light, far object. So don't forget that if you are talking about accommodation, you talk about the sacral muscle, the ciliary muscles, suspensory ligament, and the lens. And when you're talking about the pyrale mechanism, talk about the sacral muscle, radial muscles, and then the iris. All right, now let's start to answer the question. Name the process that occurs. When the curvature of the lens changes to focus a near or a distant object. Definitely, how do you call that? We call that accommodation. We call that what? Accommodation. So it means that the answer here is going to be accommodation. It's going to be accommodation. And then they are saying that the pupil size to regulate the amount of light. If they are talking about the pupil size, then it's going to be pupillary mechanism. Pupillary mechanism. Then they are saying that give the letter of two diagram A, B, C, or D. A, B, C, or D. That represents what? It represents in dim light. Dim light is gonna be B, and D. Why B and D? Is because if you look at this space and this space, they are big, while this space and this space, they are small. So here, more light enters, more light enters here. So it means that this is dim light, this is dim light, also this is dim light. I explain. Dim light, but near object, dim light near object, dim light far object, because the lens is less convex. Okay, they're saying that give the letter of 2, so the answer is gonna be D and B. Yes, or B and D, the same thing. Then they are saying that focusing a distant object, because we are looking at the lens, the shape of the lens, so it's going to be A and B. So the answer is going to be A and B. And then they are saying that give the letter of the 2 diagram, yes, that represents the eye, whose ciliary muscles, are contracted. A and D, A, B, C, D. Ciliary muscles have contracted. So if the ciliary muscles have contracted, ciliary muscles, so the answer is going to be, when the ciliary muscles contract, what happens? When the ciliary muscles contract, yes, what happens? The suspensory ligament becomes slackened. So meaning that it becomes relaxed a bit. And then now the tension on the lens will decrease. When the tension on the lens decreases, the lens will become more convex. So now we come back here and then we see which one has the lens which is less, is more convex. Then we shall see that it's going to be C and D. So the answer is going to be C and D. The answer is going to be C and D. Answer is going to be C and D. And then radial muscles relax. Radial muscles relax. When the radial muscles relax, remember that test R with R, C with C. Radial muscles relax. Radial muscles relax. Circular muscles contract. And then since we have not mixed them up, therefore it becomes bright light. So it becomes bright light. If it becomes bright light, so what does it mean? Now it means that the diameter of the pupil becomes smaller. So I will go back and look for the diameter of the pupil when it is small. So I'll see that the diameter of pupil is small here, and the diameter of the pupil is also small here. So I'm going to say the answer is A and C. The answer is going to be A and C. Yeah, that's how you can answer such question if you know exactly. These questions cannot make you go down. Yeah, why? Because you don't need to cram. I told you in life science, we don't cram, we understand the concept. Thank you very much for joining our class. Today we are going to be looking at National Senior Certificate Examination, Life Science Paper 2, 2022. Let's go through objectives and most of the questions which are likely to have some challenges, and then we try to crush or smash those challenges so that when it's time for you to write your final exam, you can still answer these questions. Let's go straight to the objectives without wasting time. which one of the following statement is correct for oogenesis? So when you talk about oogenesis means, yeah, oogenesis means the beginning of the ovum, or the process by which ovum is being formed, or formation of the ovum in humans, or formation of the ovum. So oogenesis comes from the word oo, which means ovum, and genesis means beginning, to begin something. So it is oogenesis. So which one of the following is correct? So one diploid cell mature is one diploid. So the ovum cannot be diploid. So mentioning the word diploid makes it to be wrong. Remember, all the gametes are supposed to be haploid. It means that one N. Therefore, mentioning diploid makes the statement to be wrong. One haploid, yes, haploid mature ovum is formed. Remember, in oogenesis, let me show you here. Remember, in oogenesis, you have one cell. One cell divide to form two cells. Then the two cells, they divide to form four cells. This is the normal process of meiosis. But when it comes to oogenesis, or formation of the ovum, what happens? One cell will divide to form two cells, but this cell will degenerate. It's going to become too small. So it means that's why you call it a polar body. And then these two, also you form two here, yes, but one will not develop. So it means that it is going to become a polar body. So it means that when this one also undergo meiosis, they're going to form small cells. So these ones are just minor cells. Only one of it will develop into an ovum. That's why, and this, because it is a gamete, it's going to be haploid. That's why we say that you form a haploid, one cell, only one cell will develop, and this cell must be haploid. That is the meaning. So when we go back here, we shall see that one haploid cell, yes, one haploid mature ovum is formed, which is correct. Then they are saying that the structure in which the eye, the structure in the eye that contain high concentration of blood vessels and provide oxygen and nutrients to the retina. How do we call that structure? So if you look at the eye, the structure of the eye, yes, the dark part of the eye. Let's go to our book and see exactly what I'm trying to show you, or trying to mean. So this is the part we are talking about. This part which you see, yeah, which we call the choroid. That's the part which you're talking about. It has a lot of functions. Among the function is to have blood vessels which provide oxygen and nutrients to the retina, so that now the retina can function properly. So the answer becomes B. That part is called choroid. Then we go to another question, which is the vagina. Transports urine to the outside. Vagina, does it transport urine to the outside? No, it's not the vagina, because the outlet of the urine, for the case of the female, it is just next to the outside. It's just the outside part of the vagina. And then they're saying that it is a place where fetus develop. It can't be, otherwise you'd have seen the baby developing outside. Produce progesterone. No, vagina does not have glands which produce hormones, act as a birth canal. We usually know that the second name for the vagina is called the birth canal. Therefore, that marks it to be the correct answer. Then question 1.4, they are saying that the list below gives the characteristic of some young birds immediately after hatching. If you look at after hatching, eyes are open. Okay. They're saying can move around, cannot feed themselves. Cannot feed themselves. cannot feed themselves, no feathers. Which one of the following combination represents the characteristic of precursor? So when you talk about precursor, we are talking about when the bird is born, when it is prepared. It means that it can walk, it can fend itself, it can have all the features which can make this bird to live in environment. So you will see that because it's born prepared, the eyes must be able to see. So this point is correct. And then number 2, can move around. Even this one is correct. These two are correct. And then cannot feed them. This one is wrong. This one is also wrong. So if where I have 1 and 2, then it means that that is the correct answer. Whereby I have 1 and 2, which is C. So the correct answer is C. Then they are saying that cornea, 1.5, the cornea and the lens are both. If you look at the cornea, cornea is the outside part of the eye. If you go to our book, you see that this is what you call the cornea. You see it, the outside part of the eye, and its function is to refract light rays from the object. And then the lens is this, which is a solid part and is very important. Yeah, both of them are transparent. That's why they allow light to go through. So let's go back to our question. They're saying that cornea and the lens are both opaque. So it means that that one makes it to be ruled out. They cannot be opaque when they allow light to be to go through. Both are transparent, definitely. They are both transparent. Both are elastic and both are inelastic. The lens is elastic, but the cornea is not elastic. Therefore, it means that this one marks to be wrong, and then this one marks to be wrong. So now it becomes transparent to be the correct answer. So we give you 2 ticks there. Then they're saying that which one of the following is true for a person who has an underreactive thyroid gland. It means that the thyroid gland is not active. The thyroid gland is not active, meaning that the thyroid level will be, if the thyroid gland is not active, it means that TSH, which is being produced from the brain, yes, what will happen to it? It means that if it's inactive, it means that the TSH, the thyroxine will not be produced, because if it is inactive, then it means that the thyroid gland cannot produce thyroxine. So you have to know the function of the thyroid gland, that if the function of thyroid gland is to produce thyroxine. Yes, to produce thyroxine. So if it produces thyroxine, it means that now it is inactive. It means that no thyroxine anymore. So therefore, it means that now the thyroid stimulating, this hormone which stimulates the thyroid gland will not be produced. And then now it means that if the thyroid gland is not functioning, then it means that thyroxine will not be produced. Therefore, now we come high level and low metabolic rate. High level of thyroxine, the thyroxine level will be high. Why? Because the thyroid, the brain will keep on producing TSH to go and stimulate the thyroid gland to produce thyroxine. But unfortunately, the thyroid gland does not work. Therefore, it is going to make the thyroid stimulating hormone to be produced more, keep on producing, because there is no response. The brain keeps on producing. So the level of thyroid stimulating hormone is going to be high. However, what is the function of thyroxine? This one controls the metabolic rate. But now the thyroid gland is not working. Therefore, thyroxine is not being produced. Therefore, it means that the metabolic rate is going to be low. Why? Because we don't have thyroxine in the body. Hence, high level of thyroxine. And then, because the brain is going to keep on producing more thyroid stimulating hormone, and because the thyroid gland cannot produce thyroxine, therefore the metabolic rate is going to be low. Hence, A becomes the correct answer. And then number 7, 1.1.7 is going to be They're saying that the diagram below represents the visual defect. Visual defect with what is the visual defect? Now, if you look at this, yes, let me make it a little bit bigger so that you see it clearly. If you look at this, the image is supposed to be formed here at the retina, but now it's forming in front. So if it forms in front, yes, what does it mean? It means that this lens is too thick for the light coming from a far object to reach here, because it comes with a small beam. Therefore, it means that instead of reaching the retina, because the lens is too thick, is too convex, then the image is going to form in front of the retina. Therefore, this individual will be able to see objects which are near, yes, near, but objects which are far cannot be seen. Therefore, because you see objects which are near, we say that this person is having what you call short sight. Remember, if it is more than 6 meters, it becomes far. Less than 6 meters, it becomes near. Now, let's go to the question, because you have seen the defect. Which of the following is correct statement of the visual defect shown above? So if you look at it, they're saying that the glasses with the biconvex lenses, glasses with biconcave lenses, surgery to replace the cornea, and then surgery. What caused this? What caused this is not the cornea. What caused this is this lens. It cannot change from a more convex to a less convex. Therefore, we need lenses which is going to divert this light so that when it reaches here, it must go in this direction, in that direction which is a little bit bigger, so that now it can be, yes, brought to the retina. You see, we need so that it has a big field of view. Instead of the image forming here, then the image must form here. So what lens can do this? Now let's go to our answers, and then we see biconcave lenses, biconvex lens. Now let's look at what is a concave and what is a convex. Convex looks like this. So when the light comes, it converges it. And then concave, it looks like this. It's a diverging lens. So when light falls on it, it makes it a little bit bigger when it comes out. What do we want? Let's go back and we see. So it means that we want this light. Remember, the original light is here. So we want this light to be a little bit bigger. So it means that we're going to put a lens here, so that when the light comes like this, instead of taking this route, it's going to take this route. So it is a little bit diverged. And this lens is called a concave. Yeah, it's called the concave lens. Therefore, we go to the question and now see where is the concave lens. It is here, but they are saying bi. Yes, it is bi because this side is concave and this side is concave, hence to become the word biconcave lens. Yes, so the answer becomes B. Then we go to 1.8. The list of some component of the nervous system is provided below. Then they are saying that the brain, the cranial nerves, the spinal nerves, and then the spinal cord. Which one of the following combination applies to the central nervous system? Central nervous system is composed of two things, that is the brain and the spinal cord. So you look for where there is the brain and where there is the spinal cord. So let's go to where there is a brain. The brain is part 2, part 1, and then the spinal cord is part 4. So you go for 1 and 4. 1 and 4 only, which is B. And then we go to what about these other ones? These other ones, they belong to what you call the peripheral nervous system, the nervous system of the sides.